Good morning. How y'all doing? Let's look at this one. Hi, good morning. All right, cool. It's 9 o'clock. All right, so hold on to the seat of your pants. We're gonna get going. Please slow me down if I'm going too fast at any point. But we have a lot to get through and very little time to get through it. So, without further ado, yesterday, what we went through was basically a flow chart of the anatomy. And as we were going through the flow chart of the anatomy, we stuck primarily to the GI tract, the gastrointestinal tract, because the GI tract is this tube that just runs all the way from mouth to anus. Bring that up at your next Thanksgiving dinner. It's quite an interesting topic. Um, it's true, and it's got a few sphincters in between, and it's going to have one valve that you guys have to learn about, and that's it. Just one big long tube. And what's so interesting about the tube is that the tube is going to be comprised of different structures. And so as you go along the tube, well, needless to say, structure determines function. So the function is going to change as we move through this tube. Everybody feel pretty comfortable with this idea so far? I'm sure the answer is yes. Now, there is a general structure of the entire GI tract, and that's what's on your screen right now. Just like the respiratory system, and just like every single system we've talked about, cardio system, even bones, these organs have layers. Okay, just like ogres have layers, these organs and GI tracts and respiratory tracts, these guys have layers. And we can categorize these layers relatively easily. The first layer is always called the mucosal layer, mucosa. And the reason why is because this is where mucus is made, and almost the entire body has a layer of cells that can somehow make or move or process mucus. And so that innermost layer, the layer that's right next to the lumen, it's called the apical layer, right? That apical surface and the basal surface technically, but that entire epithelial layer is part of the mucosa. So if you look at the screen right here, you're going to see, maybe my mouse, that right here is pointing at, boom, this little bracketed area. This is the mucosa layer. So if you guys in lab are looking for the mucosa layer, this is what you're looking for. The mucosa layer will be comprised of the epithelium, where the epithelium is connected. Okay, so whether it's a simple columnar epithelial tissue or a stratified squamous epithelial tissue, it just depends on where we are in the system, right? Well, where it's all going to be connected is called lamina propria. And then finally, underneath this is called the muscularis mucosa. Muscularis, what does that tell us? What is this layer going to be comprised of? It's going to be muscles. Now, it's a very thin layer. I'm hoping you guys can see what we're referring to. Literally, it's just the tip of the arrow. That arrow turned out to be huge, but still, I hope you guys get the point here. It's just the very tip of the arrow. There's this dark band right there. That's it. It's very, very thin layer of smooth muscle. So is this going to be causing contraction and relaxation of these gigantic GI tracts? No, that's not what this muscle here would be designated for. This muscle here is for small movements of the epithelial tissue. Does that make sense? The epithelial tissue can move, it can do things, and it needs contraction and relaxation for that function, just very small movements. And it's the muscularis mucosa that does that. Now, the next layer down, you got mucosa, and then you got submucosa. Below the mucosa is submucosa, easy peasy lemon squeezy. It's going to be comprised of glands, even though those aren't listed here, and the submucosal nerve plexus. Nerve plexus. Remember, everything that we end up talking about has intrinsic and extrinsic controls. True story? And so if I'm going to have an extrinsic control, I better have some nerves that communicate with it. And that's what the submucosal nerve plexus does, is this is going to communicate with the muscularis mucosa and tell it what to do. Make sense? Perfect. The third layer out is called the muscularis externa. The muscularis externa, this is a layer that I always ask you guys for on a lab practical. And I'll say, what is this specific structure and what's the general structure? So the general structure, it will be muscularis externa. Now within the muscularis externa, there are actually multiple layers of muscle because structure determines function. So for instance, if I have a circular layer of muscle, then it's going to be like a rubber band that circles around the tube. So what would be the function of that circular layer of muscle? Well, that's going to now contract and dilate, just like blood vessels, just like bronchioles. Make sense? So what if I have longitudinal muscles? Then that's going to shorten and lengthen the tube. Easy peasy. Now, what if I were to throw another one at you? What if I were to tell you that in one of the organs of the GI tract, there's an additional layer of muscle that's oblique? What does that word oblique tell you? Anybody remember? Yeah, diagonal or angled. Good. And so now if I start contracting a diagonal muscle, an angled muscle, what's it going to do to the tube? Yeah, bend and twist might be a better term for it, right? It's going to literally twist that particular organ, and that's going to be your stomach. So I hope that makes sense, that your stomach is going to have an additional layer of muscle. But if you're looking at the entire GI tract, these two layers, the circular and longitudinal layers, they're found through the entire GI tract. So this particular image is just this similarities between the entire system. Make sense? So right in the middle, sandwiched between the circular and longitudinal layers, there's a myenteric nerve plexus. Well, again, we're going to have extrinsic controls of muscle contraction and relaxation. I better have a nerve plexus, a highway. Plexus means network. So I like to think of it as a highway, but it's a network of nerves that communicate with the smooth muscle. This is going to be where our autonomic nervous system comes into play, because these are smooth muscles, so they need autonomic nervous system control. Finally, the outside layer is just connective tissue, help holding everything together. This is generally made up of serous membrane. You guys remember that serous membrane because we talked about it around the heart, we talked about it around the lungs. Around the heart, it was called pericardium. Around the lungs, it was called pleura. And now, when we're down in the digestive system, most of the time it's going to be called peritoneum. And since it's directly attached to the organ, it would be visceral peritoneum. However, Here's the trick, is we're not always inside of a peritoneal cavity. If you don't have a peritoneal cavity, we end up calling it adventitia, which is the term that you learned for the connective tissue surrounding the respiratory system. So it's a little bit of a pain in the patella. I will end up showing you guys a different picture down here to help you understand this a little better. So what we have going on with that outside layer is that the peritoneum is a lot more complex than the pleura and the pericardium because the way the abdominal cavity is set up. So if you look at the red line here in this picture, the red line is a representation of the parietal peritoneum. So it is attached to, it lines the abdominal cavity. Everybody see how that works, right? Now, what I want to remind you guys of, the visceral peritoneum, visceral pericardium, visceral pleura, is that it's like a hand getting pushed into a balloon. So imagine the liver here is a gigantic hand getting pushed into the balloon. It's going to take a part of that peritoneum and adhere itself to it all the way down. Now, the tricky part is now we got large intestines and small intestines that are wrapping back and forth and back and forth and back and forth. And that visceral peritoneum follows it all along. However, do you guys notice something strange about back here? Are there organs back here that are not inside of the peritoneum? They're not actually associated with it. And this is the pancreas, this is portions of the duodenum, this is portions of the sigmoidal colon, and the rectum are all back here as well. Okay, these organs are not inside of the peritoneum, but they're still generally part of the digestive system. So if it's inside of the peritoneum, where we have peritoneal cavity and everything, then the outside layer, I know this is weird, sorry, then the outside layer is called visceral peritoneum. But if it does not have any portion of it that's associated with the peritoneum, then we call it adventitia. I know. You guys got this though. So something like the duodenum would have adventitia, whereas something like the transverse colon would have visceral peritoneum. And we've got a whole bunch of images to kind of walk you guys around this. Intraperitoneal means that it's inside of the peritoneum, and if that's the case, then there's visceral peritoneum and parietal peritoneum. But what if it's behind the peritoneum, like we just saw with this picture? There's some organs behind it. Well, this is called retro, behind, right? If you're wearing retro clothes, it means that you're wearing clothes that are, you know, maybe a little bit, you know, behind current times, but most likely coming back into style, because why the hell not? So retroperitoneal means behind the peritoneum. This is where we come across organs like the duodenum and the pancreas, portions of the descending colon, but not the whole descending colon. What a pain in the patella that is. But the ones that I'll test you guys on the most would be something like the duodenum and the pancreas. Those are retroperitoneal. So what do we have surrounding the duodenum and the pancreas? Do we have visceral peritoneum, or do we have adventitia? Seems like you guys are getting it. Adventitia. Awesome. Good job. Good job. So there's a few other organs that are like this. I don't know how this got moved all the way down here, but it is what it is. There's a slide that kind of walks you guys through, you know, what's retroperitoneal versus non-retroperitoneal, and what's ventral, what's dorsal. I won't test you guys on all of this, so don't panic, but it's a really good representation of what's visceral peritoneum, what's parietal peritoneum, And then the last thing I'm going to point out here is, do you guys notice that the peritoneal cavity is a lot bigger than the pleural cavity? It's a lot bigger than the pericardium. Yes, no, maybe so? Not to mention that your intestines are relatively mobile. They're on kind of this weird sheet, like it's almost like a clothesline, with your intestines wrapping back and forth between something called the mesentery inside of all of this. It's really cool, the mesentery. You guys will be seeing a little bit more of the mesentery in lab. But what you can do with the patient is you can actually sit them in a way, don't sit them, lay them down in a way that you can push this portion of the intestines up, and it makes the peritoneal cavity even bigger. And most people have heard of different types of injections. There's intramuscular injections, intravenous injections, but there's also an intraperitoneal injection that some of you guys will use in the future. You will be injecting certain chemicals, drugs, into the patient's peritoneal cavity. Melissa, this is primarily used in chemotherapy. And for anybody else that's interested in cancer treatments and stuff, a lot of chemotherapy is going to be injected intraperitoneal. So it's a pretty cool structure to think about, and I tend to find a way to test on it, especially in the lab practical. All right, so how does this help us with the rest of the system? Well, it definitely does, because as we're thinking about structures like the salivary glands, right? They are an accessory organ that is going to be pushing materials into the oral cavity, pushing saliva into the oral cavity. But the oral cavity is part of the GI tract, so it must have some it must follow. How about that? What we just learned about this. So for instance, the mucosal layer, there's a mucosal layer inside of the mouth and at the back of the throat. So let's think about the type of epithelium that might be found in the mouth. Remember your epithelial tissue. You got simple squamous, stratified squamous, simple cuboidal, simple columnar, pseudostratified columnar. Those are the main ones, and then transitional down in the bladder. Of those different types of epithelial tissues, which would be most advantageous to have inside of your mouth? The back of your throat. Imagine eating Doritos. That's always my go-to, because Doritos are sharp and crazy and abrasive. Do you want a single piece of paper, or do you want a ream of paper? For everybody that's paying attention, do you want a very thin piece of paper with very sensitive cells, or do you want a massive amount of thick ream of paper? I mean, imagine tearing a single piece of paper. Just pick up 10 pieces of paper and see how easy it is to rip. It's not going to be very easy to rip. So there's always some logic that can go behind what type of epithelial tissue do we have in the system. So mouth is going to be stratified squamous. What about the oropharynx? So if we're talking about the back of the throat, are we swallowing food here? Yes, we are. So what sort of epithelial tissue do you guys think you're going to find in the oropharynx? Well, there we go. Stratified squamous. What about the laryngopharynx? Food's still passing by here. Stratified squamous. What about the esophagus? Massive amounts of food passing down through the esophagus. Stratified squamous. Does that make sense to everybody? And I'm trying to do a combo lecture and lab here at the same time. So you guys are getting this combination of information. But like with the GI tract, if we go in to look at the esophagus, what you'll notice about the esophagus, and you won't have to look at the esophagus for lab. No cilia, definitely no cilia. Cilia are a very fine hair-like structure. Do you want food rubbing down cilia? Like, imagine again this bolus of Doritos, right? Corn nuts. Passing down, it would break all those cilia off. So that's not something that we have in place. Cilia is for mucus inside of like a respiratory system. Good, good, good. But now look at this, right? You're not going to need to look at the esophagus for your lab practical. That's not the point. The point is if you're starting to look at this now, oh, come on, stop doing this weird Zoom thing. Everything likes to dance here. OMG. All right, maybe we do this way. All right, let's try this again. Hey, all right. So remember our different layers that we just walked through, and maybe you'll start to see them here, right? This first layer, mucosal layer. Pretty cool, huh? The next, I mean, it's very obvious. The next layer below that, submucosal layer. And then we get muscles. And the way the direction that the muscles is actually pointing changes the color that you're going to see in those muscles. So the esophagus is going to have an inner circular layer and an outer longitudinal layer. Make sense, everybody? So circular layers and longitudinal layers, and you could see everything that's in here. But going back to the point of the epithelial tissue, do you guys see how thick this is? Right? This is stratified squamous all the way down. So many different cell layers. All of that big, thick, protective layer. Make sense? So what's it gonna look like? And this is something you have to look at for lab. What's it gonna look like if I go from the esophagus to the stomach? Now the stomach completely changes its function. By this time, we're getting a bolus of food down into dropping it into a pool of acid. We're gonna get there in just a second. But what I want you guys to notice is just a little bit confusing because of the way it is, but on the right, what type of epithelial tissue do you guys see over here? What do you guys see? So we just looked at it. How many layers of, remember, these are nuclei here. So 1, 2, 3, we're finally losing track. 50, 60, 70, 1 million. We have multiple layers of cells over here. So what type of tissue is this? Stratified squamous. It changes. Stratified squamous just abruptly ends, and now we're going to get down to a simple cuboidal, just packed full of goblet cells. All of these little round circles right here are goblet cells, so secreting massive amounts of mucus. Simple columnar, massive amounts of mucus. Oh my gosh, that's the stomach. All right, structure determines function. So the structure abruptly changes. That means I'm going from the esophagus and it just emptied into the stomach right there. I think that's pretty cool. This is the gastroesophageal junction, and you will need to look at this for your lab practical. And I'm trying to tie it in with your lecture because of the GI wall. And now, as I start talking about saliva entering the system, chewing food, swallowing food. Hopefully you guys start to think about what type of epithelial tissues are found in each one of these locations. Can you guys try to work on that for me, pretty please? Okay. One of the next tough subjects comes because of the 6 different processes. So I need you guys, I'm going to just start a whole new page for this because of how important it is and how semester after semester after semester, students just can't seem to follow me on this, even though I point it out right here. Let's try to remember if I have a cell, we tend to draw simple cuboidal cells, even though a lot of the digestive system is simple columnar. It doesn't matter. It's all just a representation of that system. And if you're looking underneath a microscope, you're going to see a nucleus. Can't really see the cell wall borders or anything along those lines, but it's there. Now, on one side of the epithelial layer of the epithelial cells is the opening of that tube, which is called a lumen. Yes, no, maybe so? The lumen is the inside of that tube. All tubes, whether it's plumbing in your wall, the inside of a tube is called a lumen. Whether it's a straw, you got the lumen of a straw. And the surface that comes into contact with the lumen is called an apical surface. Apical surface. So from here on out, if you guys see a homework problem or a test problem, and I use the word apical, I do expect you guys to know that word. That's the surface of your epithelial tissue that comes into contact with the lumen. These epithelial tissues, these epithelial cells, are all connected to connective tissue. And they're connected at a basement membrane. If you guys remember that from A&P1 or not, I don't know. This is the basement membrane, okay? A lot of times we call this the lamina propria, if that's what you learned. But I like using the word basement membrane because the surface of the cell that's attached to that basement membrane is called the basal surface. So if on a test I'm talking about a basal surface, you guys now have that. Lateral is over here on the side, that's the lateral surface. Often these two words, basal and lateral, will just get combined together. We call it the basolateral surface, and then we have the apical surface. Apical surface comes into contact with the lumen, and so the structures on the apical surface are going to be unique because of how they are interacting with the lumen, versus the basal surface, the structures down there will be unique because it's interacting with that connective tissue down below. And if I need to write this in to remind you guys, then I will. Don't forget, this is where we find interstitial fluid. And then down here somewhere, we're going to have plasma. Does that make sense? This is connective tissue with interstitial fluid. Typically, it's going to be some form of an loose areolar connective tissue, so lots of fluid can move around in there. Awesome. Now, the part that students, well, besides this, you know, this is boring. It's a little dry. I mean, I must admit, I get that. So students don't want to focus on this, but you must learn this. What the drawing that I just drew, you should practice. Because this drawing you just drew is going to be the rest of your semester. Between digestive system and the urinary system, a big part of the function of these two systems is related to taking something from the lumen, moving it into the cell or in between the cells. That's always a possibility as well. Through the cell and getting it into the bloodstream. This is a process called absorption. If I'm moving it from the lumen into the blood, then I am absorbing that material. Yes, no, maybe so? Now, what if the cells are making something? Then the cells need materials from out here. So they're going to be taking materials sometimes from the blood. Sometimes they're just taking materials from the IF, and then they make something that needs to go out into the lumen. So now I'm going to take this material and I'm going to move it out into the lumen. What do I call this? This is This is secretion. It's not excretion yet. Excretion means removal from the body. So sometimes it will be excreted if it goes into specific lumens that allow for excretion. Does that make sense, Tariq? And everybody else? So this is secretion. So right now I got two words. I got absorption and I got secretion. Sounds like those are two processes of the digestive system. True story? You guys are going to have to learn all about these. But the most important thing to do is to find a foundational knowledge. If we're talking about absorption, and if we're talking about secretion, they have something in common. And that something in common is that they're going to be moving material across the cell membrane. They have to move it across cell membrane and then move it across the cell membrane. They have to move it across cell membranes and then move it across the cell membrane. And is that a magical moment? Does it just magically cross cell membranes? If I have something like glucose, does it just magically? Obviously, the answer is no, because I'm being sarcastic with the whole magic thing. How the heck am I going to get all of this material across cell membranes? Whether or not it's, an amino acid, whether it's a glucose, whether it's ions and water. How do I get things moving across cell membranes in the human body? This is one I could wait on all day. All right. So Ashley got the word that I was looking for. Great job. Everybody else has a good, basically a part of the answer. So for instance, what do I mean by that? Parker put gradients. I love gradients. Everything in the universe is trying to go from high to low, but not everything in the universe can go from high to low. High to low tells you passive. Andy put active transport. Well, that tells me I'm taking something from low to high, but not everything in your body can move from low to high. We only have certain active transporters out there. The combination of those two, passive transporters and active transporters, all I'm looking for is a transport protein. Does that make sense? If I'm going to move something across the cell membrane, I need a transport protein. That could be a channel, for Pete's sake. Or it can be some form of transporter, a facilitated diffusion, or an active transporter. No, Tariq. No. These are proteins that are made by the cells and inserted into the cell membranes. I love where you went. Liver's powerful, but it's not that powerful. Thank goodness. So here's the moral of my story here. This is what I just spent the longest time this particular lecture on anything, is that if I mention the word absorption on an exam, just in lecture, if I mention the word secretion on an exam, lecture, homework, doesn't really matter, what I'm talking about are transporters. You must learn the different transporters that are involved in the absorption and secretion of different materials, because structure determines function. If I'm secreting saliva, I'm going to have different transporters than if I'm secreting sodium bicarbonate. Does that make sense? And the final nail in the coffin is that when you give a patient a drug to try to help them, hopefully, again, those drugs, they're not magic. They're acting on things in the human body, and most drugs act on proteins. And now what we're dealing with is when we give a patient a drug, most drugs are going to somehow act on transporters in the body. That's what they do. Whether it's direct interaction with the transporter or whether it's acting on some downstream process. So it's either going to be a transporter or a receptor that drugs are acting on. It's a pretty straightforward concept when you start to think about it like that. So Tariq, you're not wrong, secretion from plasma and illuminant. I might have actually said that before in the past, like just like that, but it's not. Secretion's something being made by the cell. The cell needs products to make what it needs to make. So it's going to pull those products from the plasma or from the interstitial fluid into the cell, then makes it through a chemical process, and then it secretes it. So technically, secretion's coming from the cell to the lumen. Does that make sense? I'm not trying to be confusing. I'm just trying to clarify. All right. So if I talk about absorption, what should you guys be thinking about? If I ask you any question from here to the end of the semester about absorption, what should you guys be thinking about? A transporter. You're going to have to know what that transporter is. We'll learn the different transporters. And a lot of them you've already learned. So that's the good news. What about if I talk about secretion from here to the end of the semester? What should you be thinking about? Just getting this out there so you guys are aware. Transporter. Awesome. So let's get back to the lecture. And so now if I'm in here in the oral cavity, one of the processes that acts in the oral cavity is the secretion of saliva. So yeah, we learned a lot about saliva. We learned how much we make every day. We learned that the medulla oblongata has a salivary nuclei. Awesome. That's controlling part of the autonomic nervous system. Fantastic. But saliva is secretion, isn't it? So the next slide is how do we go about making the large majority of our saliva? And the large majority of our saliva is just water. Okay? It's just going to be a salt water. So let's talk about how the body, the entire body, this is not just salivary glands. These are colonic crypt cells. These are actually how portions of your respiratory system are going to end up producing different types of mucus. They use a process similar to what's on the screen right now. And what I'll point out with most of my pictures is one side is going to be the lumen, the other side will be the interstitial fluid. So if I'm looking at these cells, what surface, it's gross. What surface am I pointing to right now? That's an apical surface. Awesome. Now, what surface am I pointing to now? Cool. Awesome. Now, what I want to get you guys to think about is when you're looking at these pictures, figure out what the final product is going to be. So the final product in this particular picture, it's not super clear to students at first, but we're trying to get things into the lumen, so we're secreting. And what we're trying to secrete is a salt water, which means sodium chloride, that's just your table salt, and water. So what products do we need in the lumen? We need sodium, we need chloride, and we need water. Does that make sense so far? That's what I mean by look at the end of the story. What are we trying to get? We're trying to get sodium, we're trying to get chloride, and we're trying to get water. All right, cool. So how do we go about learning this? There are two different ways to go about learning this. One is just to sit down, draw this, and try to memorize it. I like that way, but I think that there's a little table that you guys can learn that will help you as you're studying digestive system and renal system. And this table is just going to be where you write down apical and you write down basal. And then over here in the very first column, you're going to be talking about what system that we want to be studying. So in this particular case, we're going to be talking about salt water. So sodium, chloride, and water. So this is how the whole body is going to do sodium chloride water. So then you'll have a table, and in this table, you're going to say what transporters are found on the basal surface and what transporters are found on the apical surface. So it's just a way to organize all the transporters that you're going to be learning from here on out. And then you'll be able to take and draw a diagram like this. Whether or not you draw it up and down like this, or whether you draw it sideways, like what's in my lecture presentation, right? Over here is basal surface. This is going to be the IF. Over here is going to be the lumen. So we got a basal surface and we have an apical surface. Make sense? And actually, I'm going to split these guys in half here because I want a little bit of room to play with. So just 2 cells. That's all I did there. So what I'm going to teach you guys is when you have something like this, and we start to think about what's on the basal layer versus the apical layer, I always try to put the most relevant transporter first. And since I'm trying to get sodium chloride and water from over here to over here, I'm just going to draw my transporter on that basal surface. And I need to get materials from the interstitial fluid, from the plasma, and I need to bring it into the cell. So it's just a thought process to get you started. What do I need? I need sodium. I need chloride. But we don't have transporters for just sodium chloride in the human body. What we have is we have transporters for sodium, chloride, and potassium. It's literally called the sodium 2-chloride potassium symporter. Okay, symport means that it's moving these guys all in the same direction. This is a brand new transporter. You guys have never learned about this one before. This will be the first time you see it, but not the last time you see it. Sodium 2-chloride potassium symporter. What do you know it is moving? Sodium, 2 chlorides, and 1 potassium. Symporter means it's moving it all in the same direction. If it was antiporter, it would mean that it's moving things in opposite directions. Make sense? So what's going inside of the cell? Well, we have potassium. I got a couple of chlorides. And I got a sodium. That's getting moved in using this symporter, the sodium 2-chloride potassium symporter. Do we want sodium levels to be high inside of cells? Just go with the same foundation of knowledge that you guys have learned in the past. The answer is no. So we always want to try to get that sodium back out. We always want to try to get that sodium back out because sodium high inside of the cell is bad for the resting membrane potential. So sodium, we want to move back out. Now, I know we want sodium over here in the lumen, but oddly enough, that's not how the body works. It's got one thing that's in place to be moving sodium all the time. And that's the sodium potassium pump. If I'm going to move sodium, I want to use ATP to move sodium out. So I'm going to move sodium back out. But what's going to get transported into the cell at the same time if I'm using a sodium potassium ATP pump? So now I'm moving more potassium into this cell. So let's go ahead. Over here on our basal surface, we have a sodium potassium pump. The point of this is to get the sodium back out of the cell. But now we have a lot of potassium inside this cell, don't we? Just one step at a time, peeps. That's the great thing about just slowing yourself, thinking this as a flow. Now we have potassium here and we have a potassium here. But all cells in the human body have a lot of potassium leak channels. The potassium leak channels determine the resting membrane potential for these cells. So the good news is we've already learned about these. When we were learning about all action potentials and stuff like that, how do we get potassium out of cells? We just let it leak back out. Does that make sense? So over here on the basal surface, we're going to have a potassium leak channel. Awesome. So all the potassium that's coming in, we're just going to leak it back out. The sodium that's coming in, we're pumping it back out. Now we're left with chloride. So at this point, the only ion that is high inside of the cell, besides potassium, because it's always higher inside the cell, but that's a whole nother story, is chloride. Where do I want this chloride to go? I want this chloride going over here into the lumen. Chloride's building up inside of the cell. Now I'm going to be able to move chloride from an area of high to low. And how can I do that? I got a chloride leak channel. That chloride leak channel is going to allow all this chloride just to leak through that cell into the lumen of this tube. Is everybody with me so far on the different transporters? We start with the sodium 2-chloride potassium symporter. Sodium's building up inside the cell. We don't want that, so we pump it back out. That brings another potassium into the cell. It's okay, we want potassium high inside the cell, but it's gonna go through its little homeostasis and potassium's gonna leak back out. By the way, what would happen if we put the potassium leak channel over here instead? What's gonna start building up inside the lumen of the tube if the potassium leak channel was over here? We're gonna start getting high levels of potassium inside the lumen. We don't want that. And that's why it's important to make note that on the basal surface is the potassium leak channel, not the apical surface. It completely, structure determines function. There are other places in the body where the potassium leak channel is over here on the apical surface. There are other places in the body where that happens. And that is going to change the function of that particular organ. Does that make sense? Just where we're placing these different transporters completely changes the function of that system. Awesome. So we're almost there. We're almost there. Because what's building up inside of the lumen of this tube? Lots and lots of chloride. So the chloride concentration's going up and up and up. What charge does chloride have? A negative charge. And what does negative charges attract? Don't go straight to hydrogens. Negative charges attract positive charges, right? They're magnets. The world is all about negative attracting positives. Right now we have massive amounts of negative charge building up. Electronegativity. That's what's going on over here. There's a whole bunch of electronegativity. What's building up over here? Lots and lots of sodium. Positive charges, negative charges. And as these negative charges and the electronegativity inside the lumen builds up, it's going to be like a magnet. It's going to be saying, hey, positive charges, come this way. And the positive charges here are like, I can't get through the cell. There's no transporters for me. And so they get sneaky. So instead of going through the cells, they go between the cells. Because remember, there's a space between the cells where there's desmosomes, tight junctions, and that sort of stuff, right? This is called para, around, cellular transport. So paracellular transport, due to the electronegative gradient, due to this massive amounts of electronegativity, it's going to attract sodium, and sodium's only got one way to get there, and that's in between the cells. It gets all sneaky, and it just moves, due to its attraction to the negative charge, it moves between the cells. Everybody with me? Where does water want to be? Party! And as the party moves, this party's moving. Sodium's the partium, yo. As that party moves, water's like, no, you can't go without me. And water moves with it due to osmosis. Now we got sodium chloride water. Does that make sense? You gotta slow down when you're going through these different transport mechanisms. You gotta slow down and try to draw it out step by step. If you just look at the final product, it is very confusing. If you just look at the final product, it's pretty darn confusing. But now if you look at this, step 1, sodium chloride and potassium all enter through this importer. Potassium leaks right back out. Sodium gets pumped back out and exchanges for potassium, but potassium just leaks right back out. The apical surface has a chloride leak channel. If this chloride leak channel was over here on the basal surface, chloride would just move right back out to the IF. We don't want that. We need chloride to build up inside the lumen. As it builds up, the electronegative charge is building up, and that is going to attract sodium paracellular pathway. Water just has to be where the party's at, therefore water follows. You guys feel okay about this? Awesome. Now, as you guys are going through and studying, what you're going to find is a few more of these pictures. They all come from slightly different sources. So the general gist is always going to be the same, though. Is that one side's going to be lumen, so there's going to be an apical surface. The other side's going to be interstitial fluid, so there's your basal surface. And if you go to your table and you fill out that table, you'll learn what all those transporters are. And then you start walking through this step by step. So for instance, here, we're trying to secrete a bicarbonate. Don't write any of this down right now because I have to get back to the rest of the lecture. But if I'm secreting bicarbonate, then I need to make sure that I have the products for bicarbonate. And how do we know the body's always making bicarb? This should have been part of that respiratory knowledge. I'm hoping that it's just a base part of your knowledge now. How do we make bicarb? We got to make it from carbonic acid. And where does carbonic acid come from, y'all? What molecule is critical, is the most important molecule for making acid in the human body? It produces over 70% of all the acid in your body. That's CO2. And so in this particular cell, the first product is CO2. And as CO2 enters the cell, well, shit, this particular cell has lots of carbonic anhydrase, just like red blood cells did, just like the CSF did. So CO2 gets converted in the presence of water to carbonic acid, which now dissociates into hydrogens and bicarb. Now we got our bicarb. Let's move the bicarb out. We got some byproducts we got to deal with. Let's move them out. And then there's some other transporters that are going to be helpful in this process. Does that make sense? Again, you don't need to understand every step of this so far. I just want you guys to see that no matter which process, whether absorption or secretion, there will be similarities, and they're going to be very confusing at first. And the more you do them, the better off you'll be. Moving on. You guys are going to need to learn this swallowing reflex, but don't overthink it. I don't test on it super heavily. It is important. The process of swallowing starts voluntary. You're chewing, you voluntarily swallow, and everything else after that is involuntary. So what nervous system do we know is controlling the pharyngeal phase and the esophageal phase? If it's involuntary, what nervous system is going to be doing this? Tariq, you did it again. I love you, man. The autonomic nervous system. The autonomic nervous system. And it's pretty simple, really, when it comes down to it. The second that bolus of food, this is called a bolus, is you're chewing, right? A big clump of food that you're chewing, it's mixing with the mucus, it's getting salivary enzymes in there, and some lipases, but the lipases aren't functional yet. This bolus hits the oropharynx, and the second it hits the oropharynx, it's going to start that pharyngeal phase. And this is going to be controlled by a number of different nerves that you guys aren't responsible for, but they're cranial nerves. You know, that if those cranial nerves were damaged for any reason at all, then we're going to start to have issues, like the glossopharyngeal nerve. Swallowing can be disrupted if there's damage to some of those cranial nerves. But nonetheless, the bolus of food and the depression of the tongue is going to close that epiglottis down over that laryngeal opening, the glottis. Good. We don't want food going down in the larynx. And it's going to relax this upper esophageal sphincter. So it's going to relax the sphincter, which is right here. It allows the bolus to enter the esophagus. As soon as the bolus enters the esophagus, that esophageal sphincter is going to close. Sweet. Now what you'll notice about the bolus of food is that it's going to, of course, pass down into the stomach, but it does it through rhythmic, like, contractions and relaxation of that esophagus. Do you see how where the food is not, it's contracted, and where the food is, it's relaxed? Yes, no, maybe so? This process is called peristalsis. This is a term you must know for the exam, peristalsis. The way it works is actually the smooth muscle and that mucosal layer of the esophagus. It's really smart, for lack of a better word. It can recognize exactly the different pressure gradients that are being put on it. So as a bolus of food is being passed down, as the front of the bolus of food comes into contact with the walls of the esophagus recognizes that through some baroreceptors, some pressure receptors. And what it does is it relaxes the muscles at the front of the bolus. And now the muscles can recognize where the bolus is at, and as the pressure diminishes again, behind the bolus, those muscles will contract. And so the muscle contracts right there, and then the muscle's going to contract right there, and then the muscle's going to contract right there. And what's it going to be doing to that bolus of food the entire time? Right? It's just pushing it through. Does that make sense? Imagine a sausage link, and you just take your fingers behind that sausage link, and you're just basically pushing the sausage out of the intestinal lining. Pretty cool. Yeah, the casing, exactly. All right, cool. Peristalsis. That's about all you guys need to know about it. It's a mixture of the circular muscles contracting and then the longitudinal muscles relaxing and shortening at this, depending on where you're at in the system. So that muscularis external layer, that's helping with this peristaltic motion. The next slide is about segmentation. Now, let me just stop you. Segmentation does not occur in the esophagus. It's just another form of motility, so I want to point it out now. So as you guys are talking about movement or motility, which was one of the 6 processes of the digestive system, the large majority of your digestive system acts via peristalsis. Most of your GI tract is moving things through peristalsis. In the small intestines, we have another way to move material, and it's called segmentation. Segmentation is this thought process that if I have a piece of food, I'll just use a marker. If I got a piece of food, please don't need markers, and I want to get to the center of that marker, I need to digest the nutrients out of the center of that marker. My enzymes can't just penetrate this food to get to the center. So instead, I got to chop it up into tiny little pieces, and I got to mix it all together. So to just imagine a hot dog by itself sitting in enzymes. It's going to take a long time to get to the center of that hot dog. But now imagine that hot dog in a blender mixed with enzymes. Which one's going to digest faster? Obviously, it's going to be the blender. Does that make sense? So your small intestines are just a blender, and they do it by contracting, relaxing, contracting, relaxing, and as they take, and just imagine, you know, again, look at sausage, and you're just taking and squeezing different parts of the sausage, it's going to mix all of that material together. Except those, don't get your fingers in those blades though. That's where we're at. Yeah, no blades in the small intestines. It's just it's a hand mix, which is a better way to mix anyways. Doesn't heat up your food, doesn't start cooking it as you're getting it going. Cool, and that's the end of the cephalic phase, because at this point we've had segmentation, and this food is going to go down through the lower esophageal sphincter, or that cardiac sphincter, and we finally enter the stomach. So now we're going to be in the gastric phase. The stomach is not just a digester. I mentioned this yesterday, but an important part of the stomach for me is the knowledge that it is a storage container. The stomach is extremely stretchy. And that is important because, again, you don't want all of your food getting digested all at the same time. Does that make sense? I'm actually trying to look up a number. Hold about. Yeah, I was trying to look up a number. So an empty stomach is about 12 inches long by 6 inches across. But I think well, anyways, it's not telling me how big it can get when it stretches. Oh, there it is. So it starts at about 2.5 ounces, and then as it stretches, can hold up to a quart of food. That's how stretchy the stomach is. 2.5 ounces to start, and you can go all the way up to a quart. And that just depends on the person as well. It's a pretty darn important role of the stomach. And you guys are going to be learning, and actually, let me just jump to it real quick. There is an anatomy of the stomach that you guys need to know for your lab practical, for lab, that allows this to happen. The anatomy of that stomach are these gastric folds. Yeah, those hot dog. Yeah, they've trained their muscles to stretch even more. These gastric folds, these are also known as gastric rugae. Rugae is plural, ruga is singular. And I prefer ruga because I like to roll the R, it's just fun. And these are the ones I was mentioning kind of look like a bald person's head. These gastric folds are made of elastic fibers. So because of these elastic folds, it allows that stomach to be expandable. Does that make sense? There are other properties of the stomach you're going to need to learn for your lab practical. For instance, the rest of the GI tract has a circular layer of muscle and a longitudinal layer of muscle. Circular on the rest of the GI tract is always the innermost muscle layer, but in your stomach, the innermost muscle layer is oblique. And it still has a circular and longitudinal, but we add the oblique layer underneath. And what this is going to allow that stomach to do is twist and churn, twist and churn. It adds a mechanical digestion property to the stomach that wasn't or is not found anywhere else. A slight transverse action, more like a heart beating because it's twisting at an oblique angle. The fundus or opening, up here there tends to be just a bunch of empty space unless you're lying on your back. I'll show you a picture of the gastric acid inside of here. Most of the gastric acid is going to be down in the body and pylorus portions of your stomach. Two other things for your lab that you're going to need to learn about are the curvatures. There's a greater curvature of the stomach. It's bigger, it's greater. And there's a lesser curvature of the stomach. It's lesser and smaller. These are the attachment sites for the stomach. And I'll show you some of the mesentery, which is the intestinal wrapping basically that helps hold all of this together. I'll show that to you in some other pictures. Obviously, a big part of your gastric phase is digestion. We will be talking about the intrinsic and extrinsic controls of digestion of the stomach. Last but not least, it helps with protection. Everything you eat has bacteria, pathogens in it, everything. And your stomach has lots of acid, and that acid is going to start to destroy most of those bacteria. That's the good news. What else is going on inside the stomach? As I already mentioned, due to those oblique muscles, we have mechanical digestion. Many parasites can live through that. That's right, Heather. That's they've adapted, they've evolved to live in those highly acidic environments, the mechanical churning environments. But not the adults, right? The adults aren't going to be living in the stomach. They're going to make it down to the intestines. They're going to be making it down to the intestines as basically, the young stage. And then now, what's going on with chemical digestion? In your stomach, we have hydrochloric acid, and we also have something else called pepsinogen. We'll get to there in just a second, but I'm hoping you guys already say, oh, ogen. Cool. Those two things down there, they're going to help with protein digestion. Now, the lipase, perfect, Heather. The lipase that was produced in your salivary fluid, in your saliva. Do you guys remember that there was lipase that was also secreted in your saliva? And I said at the time that that lipase has no activity yet. That lipase only functions in a highly acidic environment. So the lipase from your saliva is only active in a highly acidic environment. Well, there you go. You got your stomach. So as that lipase makes it down into the stomach, the salivary lipase can finally start to digest very small amounts of fats. Make sense? Yeah, no joke, right? And some of those fats, since they are lipid soluble, can actually start to be absorbed right here in the stomach. It's still pretty tough for them, but that's this very limited absorption is happening in the stomach. By the way, alcohol is absorbed very easily, and that's why it gets absorbed here in the stomach lining. All right, there is something else being produced. You guys are going to be learning the cells that are secreting this, but it's called intrinsic factor. Has anybody ever heard of intrinsic factor before? Yeah, Heather, yeah, anybody that's done anything with like, nutrient meal planning, something along those lines, may have heard of intrinsic factor. But how about vitamin B12? Anybody heard of vitamin B12 before? Yeah, nutrition class. Ah, see, now we're on to something. Vitamin B12. Intrinsic factor is essential for vitamin B12 absorption. What about vitamin B12? Again, this is not a nutrition class. So I'm not here to try to teach you about nutrition or anything along those lines. But what I want you guys to see with vitamin B12 is that it is essential for pretty much every single human function. I don't know how else to put it, but vitamin B12 is one of the coenzymes involved in the Krebs cycle and in quite a few other forms of metabolism as well. Okay, so without vitamin B12, we're going to have some severe issues through the entire body. I'm not going to get into too much more because I'm not going to get into the whole, idea of vitamin B12 and proper brain function and organ function and all that jazz. But vitamin B12 deficiency is a pretty big deal. Can you guys just write that down? Just vitamin B12 is essential for life. Intrinsic factor is a hormone, kind of. It's a protein. And it's secreted by your stomach. And it binds to vitamin B12 that's in our diet. Vitamin B12 it's not absorbed in the stomach. In fact, vitamin B12 by itself cannot be absorbed. We have no transporters for vitamin B12, none of them, not a single one. And yet it's essential for life. So how does this all work? Intrinsic factor binds the vitamin B12. And then we have transporters for the intrinsic factor found in the small intestines. And so when vitamin B12 is bound to the intrinsic factor, then the intrinsic factor will be absorbed due to the binding of the intrinsic factor to its receptors found in the small intestines. Crazy, huh? So what happens if somebody doesn't have enough intrinsic factor? What's going to happen to vitamin B12 absorption? Increase, decrease, no change. If you have a gastric bypass, you're going to have to deal with this for the rest of your life, because what a gastric bypass surgery does is it bypasses the large majority of your stomach. That's literally why it's called gastric bypass. I'm always confused by the fact that this doesn't click for everybody. It's bypassing your stomach, in case you've never looked at this before. So a gastric bypass is going to take the tubes of your stomach and it's going to actually attach the jejunum up to the upper portion of your stomach. That's it. So now this becomes your stomach, and that's as far as it can stretch. What a great theory. It's a great idea. It does help people, but it's going to have a long-term effect on hormonal secretion. It's going to have a massive effect on hormone secretion, and your stomach produces a lot of hormones and intrinsic factors and all that sort of stuff. So now we are reliant on taking, vitamin B12, soluble vitamin B12, and dealing with all that. So for the rest of their life, they'll have other things to worry about. But intrinsic factor, I tend to test on it. No promises that I will, but it's pretty important because vitamin B12 is so critical for life. The last thing we're going to be learning about right now before break is this next thing, gastric juice. You produce about 3 liters of stomach acid every single day. That's a lot. That's going to come back into play later down the road, because if we're making 3 liters of hydrochloric acid juice, are you pooping out hydrochloric acid juice? Please tell me no. Just tell me no. So we're going to have to deal with that later. Everything that gets secreted into the system, we will need to absorb back out of the system. In addition, since it's hydrochloric acid, we don't just need to absorb it, we need to neutralize it. Does that make sense? So down road here, we're going to neutralize all this acid and we're going to absorb it all back up. What else is in gastric juice? Lots and lots of mucus and a protein called pepsinogen. And just like angiotensinogen and just like plasminogen and fibrinogen, what do we know about the ogen? Heather already wrote it earlier. Good, Ashley. We know that this is inactive. That's good. Okay, we know this is inactive. So we are going to need to activate it. So how do we go about doing this? That's where we'll go next. We're going to need to cleave it off, exactly. We're going to come back to this one. Let me come back to this one. We'll come back to that one at the end of the road. I'm actually going to save it like that. So in your stomach, we have gastric folds. And on those gastric folds, those are those ruga. So here's that stomach right here. On that stomach, we have gastric folds. And as we zoom in on the gastric folds, this is what you see right here. Maybe if I did this instead. There we go. All right. Do you guys see these big waves? These big old waves right here? Each one of these is a gastric fold. Every one of these is a gastric fold. And what you'll notice is hopefully it doesn't do what it was doing before. Nice, it's not. As I zoom in, the gastric folds, they have tiny little folds on them as well. Lots and lots of surface area. Surface area is important for many, many, many things. All right. Now, what we're going to go ahead, let me go to, this mucosal layer that you see here. What each one of these folds is, it's a gastric pit. Okay, so as I zoomed in on this, these little pits, gastric folds are covered in them. If you just look at the surface of the stomach, it looks like it's got tons of little holes in it. But those holes go down into these gastric pits. Okay, gastric pit there. Really good gastric pit right here. Gastric pit. Yes, no, maybe so. Gastric pit right here. Awesome. Now below the gastric pit, there's your epithelial tissue up there. Now what you'll notice is there's just all of this structure down here. Okay? These structures are glands. All of these structures down here are different glands. What do we know about glands? They produce hormones. Okay? Does that make sense? So that's part of that submucosal layer. Now, what you'll notice here is it's going to say mucous neck cells, parietal cells, chief cells. You guys don't need to identify these cells, chief cells and parietal cells, not in your lab practical. They are an important part of the functionality of this entire system. So let's go back to the lecture, and you can see this simplified. This right here is a gastric pit. So this is, just an animation of it. Over here, the gastric pit is just this portion, just like you just saw, just this lighter pink area. Those are the gastric pits. And down below that are the gastric glands. So in histology, all the gastric glands are down below. And when we look at the animation of it, what you'll notice is the gastric glands are made up of many different types of cells, all connected by some tubes. And those tubes, they go up into the gastric pit. Pretty simple idea right now, right? A gastric gland is made up of many different types of cells, all connected by a tube that goes up into that gastric pit. Yes, no, maybe so? Okay, I gotta wait. This is a tube. This open space right here is a tube. So the pit itself would be considered part of the tube as well. All right, cool. Now, I'm just going to get you guys up here to the gastric pit. The gastric pit has two major types of cells. Don't stress about these. I don't test on them heavily. But these are mucus cells. Mucus neck cells, surface mucus cells. What does the name tell you? The function of these cells, both the surface mucus cells and the mucus neck cells, what are they going to be making? Yeah, they're going to be making mucus. I'm not going to waste your guys' time on that. Some of that mucus down with the mucus neck cells, this is an acidic, but the large majority of the cells, and by the way, this is going to cover all the surface of the stomach as well, are covered with surface mucus cells. These guys secrete an alkaline fluid. Why do we need an alkaline mucus covering our stomach? It's a base. It's a base buffer. So what is the alkaline fluid that's being produced? What's the number one base buffer in the human body? We already talked about it today. Yes, yes. Great job, Melissa, Ruju, Andy. Awesome, awesome. The answer is bicarb. Good. Everybody, please write that down. If we're talking about an alkaline base buffer, the most likely scenarios, we're talking about bicarbonate, HCO3 minus. Is that clear? You must know that. In my class, you must know that. You must know that. It's going to come up far too often. Heather, yes. Yes, all of that all comes together. It's all part of the gastric juice. The gastric juice is a mixture of the mucus and the acid. So the key here for this particular slide is not to memorize all these cells yet. We'll get there in just a second. The key here is to make sure to understand that we're going to have hydrochloric acid produced down here in the gastric gland. And that hydrochloric acid is going to be exiting out through the gastric pit into the stomach. But acid eats proteins. Do we want that acid just to eat away the proteins of our stomach lining? No. So what does the entire surface of your stomach produce? The surface mucus cells producing an alkaline, bicarb-rich mucus that coats the stomach and protects it against the acid that it's producing. What is Tums? Tums is calcium bicarbonate. That's all it is. It's calcium bound to bicarb. So when you eat a Tum, it's going to dissociate. The calcium separates from the bicarb, and all those bicarbs that you just ate in the form of Tums now coat your stomach and help against acid reflux and an upset tummy where the acid's getting all a little bit crazy. Make sense? That's all it is. It's just bicarb. Baking soda is just bicarb. It's sodium bicarb, though, not calcium bicarb. And your body produces a lot of sodium bicarb. All right, cool. So how can we learn these different cells? I got a trick that I picked up a while back, kind of on accident, and it's really helped students out. Because just going in and memorizing parietal cell versus chief cell versus G cell, what's going on with all these things, and the order's all weird and wonky. All right, let's learn these cells in a very specific order. And let's draw out a flow chart for this. Once the cells are going to be drawn out here, I'll let you guys go on break, of course. So the first cell you guys are going to learn about is called the G cell. The next cell you guys are going to learn about is called the parietal cell. And the last cell I want you guys to learn about is called the chief cell. It's actually not the last cell. There'll be one more cell. Last cell we're going to write in over here. It's going to be called a D cell. We'll get to that one a little bit later. All right, cool. It's nice to start simple. The cell names are G cell, parietal cell, and chief cell. You already learned chief cell earlier this semester in the parathyroid gland. Remember, chief cell is not unique to a particular gland. It's just a name that we give a lot of different cells depending on the organ we're in. G cells, they secrete something. Parietal cells, they secrete something. Chief cells, they secrete something. G cells are called G cells because they secrete a hormone. This is an endocrine hormone. What does that tell us? If it's an endocrine hormone, where's it going to be secreted into? Good. Great job. So G cells secrete an endocrine hormone called gastrin. Nice. This shouldn't be too hard to remember. Proteins in the human body end in IN. This acts in the stomach, gastric, so it's just gastrin, and they're produced by a G cell where the G stands for gastrin. Parietal cells. Parietal cells, in this scheme of things right here, they're going to produce massive amounts of hydrochloric acid. Okay, parietal cells, hydrochloric acid. That's what we're going to be focusing on. The parietal cells also secrete intrinsic factor. What do the chief cells secrete? They secrete pepsin, which is just Pepsi, with an N at the end because proteins in the human body end in IN. Ogen. We already discussed this, but what do we know about the ogen? It's inactive. Nice. Now, what do we know about hormones is that a hormone needs a stimulus for its secretion. So the G cell, in order to secrete gastrin, needs a stimulus. Does that make sense? So what is the stimulus for G cells? There are two. There's an extrinsic stimulus and there's an intrinsic stimulus. Extrinsic, coming from outside, is vagal stimulation. Okay, if the vagus is stimulated for any reason, the primary reason why the vagus nerve would be stimulated is if you look at food. Yeah, good old cranial nerve 10. If you look at food, if you smell food, any of these is going to trigger the cephalic phase of rest and digest, and the vagus nerve controls over 90% of all rest and digest. So the vagus nerve is a pretty powerful nerve, and if you stimulate that nerve, it's going to cause the G cell to start to produce gastrin. Viva Las Vegas. Now, there is another way, which is intrinsic. Okay, there's another way. We don't have to have vagal stimulation, although we'll probably have vagal stimulation if we get it here. But that being said, you can just intubate a patient, for instance, and it bypasses all the vagal stimulation stuff, and it just gets food directly in that stomach. So the presence of food or stomach stretch, right? That alone could cause gastrin secretion. And then one more thing that's intrinsic, it's the presence of a particular type of food. And if you recall, I've been trying to tell you that the stomach digests a very specific type of food, a very specific type of nutrient, and it's not fat. Good try on that one though. There's a little bit of digestion of fat, very small amounts, but the large majority of chemical digestion that's occurring in the stomach is related to hydrochloric acid. What is hydrochloric acid denaturing? Cooking. Proteins. Okay, so the presence, how the hell do you spell presence? I don't even know. I didn't come close. Oh, I was close. Oh, I know, I wasn't even close. I was on it. That's a weird word. Of proteins. All right. Thank you, Tariq. I was writing it and it just didn't look right. Any of these can cause these G cells to be stimulated, which is going to then cause the secretion of gastrin. Gastrin goes out into the bloodstream, but it does not have to move far. The function of the primary function of gastrin is to communicate with these parietal cells. So gastrin gets into the bloodstream, travels very short distance, and parietal cells have receptors for gastrin. Good old Dr. Google. Well, what do we know parietal cells secrete? So due to the presence of gastrin, parietal cells are going to get activated and they're going to start to secrete hydrochloric acid and some intrinsic factor. Make sense so far? Now, hydrochloric acid goes out into the lumen of that gastric gland. So now this is going out into that gastric gland and it's starting to fill up that gastric gland. And as it does, it's going to be passing by the chief cells. And the chief cells have nerves that are highly sensitive to acid. The more acid that's in the system, the more these chief cells become activated. So as hydrochloric acid enters the system and we activate chief cells, what protein is going to be secreted? Pepsinogen. Pepsinogen. Not pepsin, pepsinogen. Now, what do we know about pepsinogen is we need to cleave that ogen off somehow. Well, the good news is we already have that in place. It's the hydrochloric acid that does that job. So as chief cells are stimulated to release pepsinogen in the presence of hydrochloric acid, that hydrochloric acid's function is to denature and cleave the ogen off of pepsinogen. Now what we're going to be left with is just pepsin. And pepsin, it has a role to digest proteins. That's what it does. It digests these big, huge, long chains of peptides. But it's not perfect. What do I mean by that? What I mean by that is proteins are made up of big, long strands of amino acids, if you guys will recall that, right? The smallest building block of protein is an amino acid. There are 20 essential amino acids in the human body, and those amino acids come together to form these proteins. Now, proteins are folded. They're folded in these beautiful different conformations. It's really cool how proteins fold. But when a protein folds due to the R group attraction between the different amino acids, what that does is, imagine a ball of yarn, or an origami or something like that. That's pretty cool how it folds together. Once it's folded together, it's functional. And once it's folded together, I can't just get to the core of that ball of yarn. What do I need to do is I need to unravel it. I need to unfold it. I need to denature it. Does that make sense? I cannot digest the core of this protein until I unfold that protein first. It's impossible. So pepsin in and of itself can't digest your proteins. It needs to have those proteins denatured first. Don't we have something in the system that helps to denature those proteins? So as you eat the protein, those proteins are denatured or unfolded. And once those proteins are denatured, now we're going to have this activated pepsin in the system, and that is what gets digested. We cannot digest a protein if there's not enough hydrochloric acid. These proteins are not chopped down into amino acids. They're chopped down into something called oligopeptides. Oligopeptides are still long strands of amino acids, sometimes, up to, 4 amino acids to 10 amino acids long still. Many. Yep, oligo means many. So we still have a number of amino acids linked together as it's leaving the stomach. So we have not completed the digestion process yet. Does that make sense? Where are we going to complete the digestion process for proteins? In the duodenum. Trying to digest proteins created. Maybe I misspoke somewhere. I'm not sure where I missaid something, Tariq. I'm so sorry. No, we're not digesting proteins created by our stomach. Only thing we do here is pepsinogen. The hydrochloric acid will cleave the O-gen off of pepsinogen. Pepsin is active in hydrochloric acid. It does not denature this protein. This is a protein that is beautifully functional in a highly acidic environment. Does that make sense? And then lipases, those are a protein. They're a protein enzyme that breaks fats apart. We have different enzymes in our body that that are activated by different pHs. And so pepsinogen activated by extremely low pH. The salivary lipases activated by a very low pH. And then there are other enzymes that only work in alkaline environments, and there's other enzymes that only act in other pH values and temperature values, and enzymes are crazy. Man, I've gone way too long here. Let me finish here. Do we want high levels of ASFIT all the time? Or do we need a way to turn this system off? We need a way to turn this system off. And we have a cell for that. It's called the D cell. Okay, this D cell is sensing hydrochloric acid. And as hydrochloric acid levels get too high, it's going to activate the D cell. And the D cell secretes a hormone that you guys learned about before called somatostatin. You may or may not remember learning about somatostatin before. I tried to point it out, but this is growth hormone inhibiting hormone. This is GHIH. So there's cells in your stomach that produce growth hormone inhibiting hormone. which makes sense because if we want to inhibit growth, do we want more proteins or less proteins available? Less. So to inhibit this system, I call somatostatin a blanket inhibitor because what's it going to do? It's a negative feedback loop. So it's going to go flat line, negative inhibition going on here, negative feedback, I guess I should say, but it's going to inhibit the G cells. It's going to inhibit parietal cells, and it's going to inhibit chief cells. It's a blanket inhibitor. It inhibits everything. So as long as somatostatin's in the system, it basically stops gastrin production, hydrochloric acid production, and pepsinogen secretion. Almost guaranteed you're going to have to draw this flowchart on the exam. Almost always make you guys do it. So think through it, start from scratch. Start just by writing the cells: G cells, parietal cells, chief cells. G cells produce gastrin, parietal cells produce hydrochloric acid, chief cells produce pepsinogen, and they link together. And now when you look at something like this, it won't be so complicated. Because when the G cells are stimulated, they secrete gastrin into the bloodstream. Bloodstream transported to the parietal cells, and they secrete hydrochloric acid and intrinsic factor. The hydrochloric acid's passing by the chief cells, causing the chief cells to secrete pepsinogen, but the pepsinogen is currently inactive. The hydrochloric acid activates the pepsinogen by cleaving off the ogen, and the hydrochloric acid is taking a big folded piece of protein and denaturing it, unfolding it, and the pepsin can come in and cleave it into oligopeptides. Oligopeptides are still multiple chains of amino acids bound together. We can't absorb these yet. We'll have to finish the job in the small intestines. Yeah, yeah, denaturing your food. Yep, yep. Because, right, that's vagal stimulation, stomach stretch, presence of those proteins in your food. It's all about the food we're eating. Good. All right, let's take a break. Let's take a decent break. Come back at 11. Yeah, exactly. Flowchart and diagram. Flowchart in and of itself, I don't think is, I mean, I'm a big time flowchart person, but I still think you need, well, we won't get into my learning techniques, but yeah, I think you need everything. I'm not a big idea of this. You must be a visual learner versus you must be blah blah learner. I just don't believe in that personally. I think you have to learn from many different angles if you truly want to learn. And the good news about flowcharts is that they're a combination of all sorts of learning. So, all right. Anywho, break. Come back at 11. Please, please, please. Still a lot left to get through today. Too much. Hello. Mm. Got a 6-hour drive ahead of me tomorrow after class. And down to South Padre with the family. I'm not looking forward to the drive. But kiddos need some beach. I don't know, Tariq. My wife booked it all. I just say yes, ma'am. I just say yes, ma'am. All right, let's go ahead and get rolling. It's gonna be lots of fun. All right, guys. So again, we are kind of sharing both lecture and lab time right now. I can prove that to you in just a minute. So let's finish up the gastric phase. Basically, at this point, we have a bolus of food down in our stomach, and we are digesting proteins in a very small amount of lipids. It looks a little bit like this. We're taking the bolus of food, gone through that the lower esophageal sphincter, the cardiac sphincter. There's that fundus of the stomach, all those gastric folds, the ruga that are, covered in these gastric pits, and the gastric pits are secreting the hydrochloric acid and the pepsinogen into that stomach. And this bolus of food is getting churned and digested. Those proteins are getting denatured, cleaved into oligopeptides. Some of those carbohydrates were digested up in the mouth, so we started getting smaller portions of carbs already, and some of the lipases are also doing their job, so they're cutting up some of the lipids. Sweet. Now our gastric juices are filled with what's called chyme. Chyme is a partially digested solid mass. Our bolus became chyme. And that chyme, it starts to build up on that inferior border of that stomach near the pylorus. Is that clear? We're down here in the pylorus. And this chyme is building up inside of this pylorus. Yes, no, maybe so? And what this is going to do, this is just a start. This is not the end of the story by any means. But as chyme builds up, it starts to put pressure against that pyloric sphincter. And the pyloric sphincter has some baroreceptors in it. And the more pressure that's put against it, the more likely this pyloric sphincter is going to relax. And as it relaxes, it's going to allow a small bolus of chyme into the duodenum, into that small intestines, the first portion of the small intestines. But immediately after a small bolus has passed through, it immediately closes again. It's going to cause retropulsion, but I never test on retropulsion. This can end up causing some other downstream effects that we're just not going to deal with in this class. There are other things that communicate with the pyloric sphincter. You guys hopefully remember me saying how smart the digestive system is. And we're going to have hormones that we're going to learn about that are secreted by the small intestines. And the hormones that are secreted down here can talk back to the stomach and say, hey, we need more of, or we need less of. We have plenty of carbs. Stop sending me carbs, right? We have plenty of proteins. Stop sending me proteins, and they can help direct traffic with this pyloric sphincter too. But the more food you eat, the more likely you're going to be causing a lot of pressure in this pyloric region to push the food out into your digestive tract down here in the small intestines, duodenum, increasing digestion, increasing the amount of nutrients going into your bloodstream. And the more nutrients in your bloodstream, the more it stores those nutrients away. All excess glucose, all excess proteins, all excess fats get stored away as fat. Your body doesn't want to get rid of it. And if you can't use it right now, it stores it away as fat for later. Make sense? So the more we put into our duodenum, the more digestion and absorption we have, the more excess energy there is, and the more fat will get stored away because your body is greedy. It wants to hold on to all sources of long-term energy because the fact that food is so freely available is relatively new in human evolution. 150 years ago, 100 years ago, heck, there's people across the world today that don't have free access to food. They don't know whether they're getting food tomorrow. So the body is still taking all excess, right, where your body would use all of it instead of not store it. Use immediately. If you have energy, six packs. Are you talking about six-pack abs? If you just eaten a small meal, like I don't know, a handful of peanuts or cashews or something like that, and then you go to exercise, for instance, right? You have not filled your stomach up. You're not triggering massive parasympathetic overload. You're just consuming a little bit of energy. Peanut butter, right? You got marathon runners all the time that have those little packs of peanut butter. Like, as they're running the marathon, they squeeze a pack of peanut butter into their mouth, and boom, now they got a free energy source. Your body is going to absorb that into the blood, and it's not going to store that energy away. It's going to use it right now. Dates, exactly. High sugar content, very high sugar content in dates. So immediate source of energy. Candy, yummy, but just it's a lot of processed sugars, and the body has a complete different function with high fructose corn syrup. Anywho, does that make sense? And we'll talk more about this. I just want to, I love the tangents. I think they're great because they are relevant, but I want to make sure that we get through everything. The next portion I'm not going to go through as thoroughly. So guys, just pay attention. It is important. You can be tested on it, but I tend not to ask you guys to draw flow charts and stuff on it. Because at this point, we're done with the stomach, and we're now putting stuff down into the intestines. What we're going to be talking about the most, which will be tested on the most with the small intestines and the duodenum, will be digestion and absorption. We're not there yet. We're just going to be talking about the intestinal phase. The intestinal phase, the first portion of that GI tract is the small intestines, and this is where the rest of digestion is occurring. Almost all digestion occurs in the small intestines. In fact, of those small intestines, I know I've mentioned this before, the duodenum does the large majority of the work. By the time that this chyme makes it to the ileum, there's almost nothing left to digest and absorb. It's almost all done already. So your large intestines are not designed for digesting food. So part of the intestinal phase, they're not designed for digesting food. They're designed to take all these liters of material you've been putting in, from the saliva to the hydrochloric acid. You're going to see a whole bunch is going to be injected into the system by the pancreas and the liver. All of this fluid, liquid material that's entering the intestines, we need to get it back out, back into our bloodstream, so we don't poop it out. That's what the large intestines is primarily for. We call this concentration of our waste. It's going to take multiple liters of material, pull out the liquid, pull out ions, pull out bicarbs. Everything we need goes back into the blood, and we only excrete what we don't need, the waste. Make sense? Large intestines also have a slightly different way of moving things. And I talked about this yesterday with motility, is they have what's called mass movements. Mass movements have to do with the anatomy of the large intestines. So let's go back a lecture and let's look at the anatomy of these large intestines real quick. This is all part of your lab. The ileum is right here at the tip of my mouse. That's the very, very end of the ileum. So that's the small intestines. And food from the ileum, chyme from the ileum, is going to enter the first portion of the large intestines, which is this enlarged region called the cecum. We're not to the ascending colon quite yet. We're right in the cecum. There's a valve right here called the ileocecal valve. Remember, a valve is just a flap of connective tissue, and it's going to allow pressure gradients to determine where this chyme goes. The valve opens into the cecum, so as the pressure is higher in the ileum, it's going to allow for that chyme to move into the cecum. The cecum houses a very large concentration of bacteria. And hanging off of the bottom of the cecum is where we find the appendix, the vermiform appendix. And anybody in here had appendicitis? Anybody in here had their appendix removed? Tariq, did you have it removed? Yeah, cool. Yeah, And Heather, I believe earlier this semester you said, hey, what's your opinion on the appendix? Well, here's my opinion on the appendix. The appendix is actually, it's, called vermiform, it's vestigial. It means it's not required for life, kind of like the gallbladder isn't either. A lot of people think that it's completely innate, that it has no functionality whatsoever. But here's the deal. That's kind of a myth. There's definitely a function of the appendix still to this date. The appendix, 1, has immune system function, and 2, it houses bacteria. It's basically like your super reserve of bacteria. Good, happy, wonderful microflora that helps keep you alive. Now, we've all been sick before. I want to say probably everybody's had gut bomb. You guys know what I'm talking about, where it cleans the system out. Yeah. It's not, no, Heather, there's a slight misconception on bacteria and where it's at. For instance, there's no bacteria up in the stomach, there's no bacteria in the duodenum, upper portions of the small intestines, there's no bacteria there. Okay, the large majority of your bacteria is in your large intestines at this point. Keep working with me though. The bacteria is found everywhere. Don't get me wrong, it's not found in the stomach, period. We're not a cow. Anywho, yeah, so the appendix stores away bacteria. So if you blow your system out, you should have a bacterial reserve down here in the cecum, but if the entire system wants to recover faster, that's something that the appendix can help with, because now we end up getting these bacterial blooms out of the appendix. There's some pretty cool papers that are written on it. Does that mean that you're going to die without it? Of course not. Plenty of people have had their appendix taken out. Does it mean that you still won't recover after you blow your system out? Of course not. People still have the cecum that houses the bacteria. So, yep. Yeah, I mean, it is what it is at this point, right? If you got appendicitis, it can kill you. So, cause sepsis and all sorts of nasty stuff, and you got to get it taken out, not to mention the pain. Now, the way that this is set up, you guys notice that the large intestines have these enlarged regions, enlarged regions, enlarged region, yeah? Each portion of this is enlarged. These are called Haustrum. It's like a German word, Haustrum. I don't know, I can't do the Haus, anyways. Haustra is plural, Haustrum is singular. So each one of these is a Haustrum. This is the motility part that I'm trying to get to. Okay, Haustrum is part of your lab objectives. The ascending, transverse, descending, and even the sigmoidal colon all have these Haustrum. And the way that mass movement works is that there's this thing called the Tene coli. See this silver gray structure that kind of holds it all together? It's the Taenia coli. Yes, no, maybe so? You have muscle groups as part of these haustrum that are kind of attached to these Taenia coli. And what will happen is that the haustrum will contract. The Taenia coli kind of keeps the entire intestines in place, but then the haustrum contracts. And as one haustrum contracts, it makes it so that all the material that was inside of it can't stay inside of it anymore, and it's going to move to the next haustrum. And then it makes another set of contractions, which now sends all of that material to the next haustrum. So from haustrum to haustrum to haustrum to haustrum. This is called mass movement, when we're moving everything from one haustrum to the next. Does that make sense? It's only one way. Due to pressure gradients, it's only one way. Yep. Due to the way that the muscles are contracting too. Just like peristalsis. Okie dokie artichokes. The other thing I want to point out in this picture while we're here, do you guys see this yellowish sheet of, it kind of looks like a membrane. It is technically a type of membrane. This is all called the mesentery. All of this is the mesentery. And there's other portions of the mesentery as well. The portion of the mesentery that holds the transverse colon together is called the transverse mesocolon, and this is a structure that you need to identify for the lab practical. So all of this connective tissue, this portion of the mesentery is called the transverse mesocolon. The rest of this we'll just call the mesentery, which I hope you guys remember that word because of the mesenteric arteries and mesenteric veins, both superior mesenteric artery and the inferior mesenteric artery down below here. Yes, no, maybe so? Cool. All right, let's move back here. That's all I really want you guys to see, mass movement. And then the next part of motility has to do with the defecation reflex, and we're going to learn about that tomorrow. But it's just baroreceptors that are going to cause defecation. Now, I know I've mentioned a number of times that almost all digestion occurs in the small intestines. By the time it makes it to the large intestines, it's pretty much done. However, there is an important thing that must get digested in the large intestines, and that's vitamin K. You have no enzymes in your body to help with the digestion of vitamin K. So you ingest vitamin K in your food, but you have no ability to digest it. However, vitamin K, does anybody remember from earlier this semester, what is vitamin K essential in? Anybody remember? It was from unit. Yes, good job. Quinn, man, that was fantastic. It's a hard one because we barely touch it. We talk about it for maybe, I don't even know, a minute or two, and that's it. Not even seconds. It's required for not hemolysis, of course, but hemostasis. But I knew that's what you knew. I know, right? This is all about blood clotting. Vitamin K is essential for blood clotting. If you don't have vitamin K, you cannot clot and you will bleed out and die. It's really that horrible. You will bleed out internally and die without vitamin K. Oh. Sounds pretty brutal, huh? It's one of the wonderful things that your bacteria do, okay? Your bacteria do what it do. One of the major things that bacteria helps with is it is able to break down vitamin K and allows vitamin K to be absorbed into your bloodstream. Without the healthy, happy bacteria, vitamin K can't get into your blood. Pretty cool, huh? I think it's cool, personally. This is what rat poison does. Rat poison blocks vitamin K absorption. No bueno. Actually causes rats to bleed out internally, hemorrhage, and die. So, yeah, wonderful stuff. Water and ions here. That also is going on in the large intestines. We have about 9 liters of liquid at this point. Bicarb rich. Remember, ions, not just sodium, not just potassium and calcium and chloride. It's bicarbs too. It's bicarbs too. Can you guys remember that for me? Please? Ions, bicarbs too, bicarbs too. I'll explain why this is important in just a second. But where does water want to be, y'all? Where the body at? Where the body at? Where the body at? And so what's going to happen in the large intestines is we're going to be absorbing massive amounts of ions, sodium and chloride, sodium down here, right? We're going to be absorbing all of these ions, just tons of them. We need to get them into the ECF. We need to get them from the lumen of the large intestines. We need to get them over here to the ECF. We're going to start to build up a lot of sodium, a lot of chloride. Chloride buildup creates a negative, electronegative charge. Potassium follows that chloride. Water, where does water want to be? Where the party's at. Yeah, exactly. Y'all, does that make sense? Without going into the nitty-gritty of this, because you guys will need to look at this. Don't stress. Don't stress. Look at all of these ions. Here's some bicarbs and stuff like that as well. We're going to be moving bicarbs into the ECF through a different mechanism. But sodiums and chlorides, sodium, chloride, all of this is getting into the ECF. So as we move these ions into the ECF, what's going to follow? This is important. Water, water, water down the board. So what would happen if we slowed down the movement of sodium into the ECF? What's going to happen to the amount of water that gets reabsorbed back into your blood? Increase, decrease, no change. It doesn't, retention's a tough one. It's retained inside the lumen of your large intestines, right? So what's going to happen to your fecal volume? All of the sodium is left here in the lumen. So where does water want to be? Water is going to stay with that sodium. Does that make sense? So what's going to happen to your fecal volume? Increase, decrease, no change. And if all of this fluid is shooting out your butt, we call this diarrhea first and foremost, but it's going out so fast that we're not going to have appropriate bicarb absorption. Bicarb is absorbed through another mechanism. So what's going to happen to the amount of bicarb that you can absorb? If everything else is moving faster, bicarb is going to have less time to go by its transporters. So what's going to happen to bicarb absorption? Increase, decrease, no change. And that is going to decrease the total available bicarb in that patient's body. Pretty big deal. Diarrhea can cause dehydration and something called acidosis. Acidosis, your body is removing bicarbs. And if you don't have enough bicarbs, then your body becomes more acidic because there's less of a base buffer available to neutralize those acids. Does that make sense? It's not going to be carbonic acid. You're overthinking. We'll get there. Hydrochloric acid. No, not that either. We'll get back to that, Tariq. That's for the renal system. I just want you guys to jump on board right now. Okay, so if we're losing bicarbs, we have less of a buffer. Yes, no, maybe so? Diarrhea is extremely dangerous. We like to laugh about diarrhea, you know, this whole thing about explosive diarrhea, and when it was at the NBC water slide show that got canceled due to explosive diarrhea on the set, you know, it's always like a gross thing. But the reality here in the medical world is diarrhea is a lot more than just gross. It's extremely dangerous. The smaller a person is, the faster they can be dehydrated and enter acidosis. So for instance, if you have a baby that has diarrhea, should that parent wait for 2 or 3 days. Be like, hey, I'll wait 2 or 3 days. I'll be fine. Baby has diarrhea, you get them to the doctor because they can dehydrate within 12 hours. They could be so sick within 24 hours that there's no turning back. Does this make sense? And it all comes about because of improper absorption of ions. Let's go one more route. Everybody's heard of a laxative before, right? Different laxatives. Milk of magnesia is one of the major laxatives. What you do is you consume the milk of magnesia, and you have no transporters for absorption of milk of magnesia. So when we make it to the large intestines, the milk of magnesia is still there. And where does water want to be? Where that party at? And the milk of magnesia is the party now because it is such massive osmolarity. Does that make sense? And therefore, you poop out the milk of magnesia and all the water goes with it. It's a laxative. Yes, no, maybe so? Other laxatives block these transporters. There's a lot of different ways that we can create a laxative, but it's all about changing the party. It's not about changing water. That's the misconception. It's all about changing the party. And once you change the party, water just go where it's supposed to go. It's very highly influenced. Get it? Got it good? Yeah, that's fine. We'll kind of skip this slide. You guys can look at this later. Yeah. So defecation reflex. Defecation reflex is all about the anatomy of the rectum and the anus. The rectum is just a storage container with a bunch of baroreceptors in it. Okay, those baroreceptors are constantly monitoring the stretch on the walls of the rectum. The anus is made up of muscles. It's the end of the road. So when I say that the GI tract is a long tube separated by a bunch of sphincters, well, the end of the road thankfully has sphincters. The end of the road actually has 2 sphincters. It's got an internal anal sphincter. Okay, internal means closer to the rectum anus surface, to the lumen. And there's an external anal sphincter, which is more lateral, and then it's just right on the very outside edge as well. You guys have those 2 anal sphincters written down? Internal anal sphincter, external anal sphincter. Now, what if I told you that the internal anal sphincter is involuntary, smooth muscle? And what if I told you that the external anal sphincter is skeletal muscle? I hope that's helpful at this point in the game. What nervous system is going to be controlling then I will believe you. That's not the point. That's not the point. The point is, what do we know about smooth muscle? What part of our nervous system controls smooth muscle? Autonomic nervous system. What part of our nervous system controls skeletal muscle? Somatic. Does that make sense? Autonomic nervous system would control the internal anal sphincter because it's smooth muscle, versus the external anal sphincter, we have somatic motor neurons down there. So the defecation reflex is kind of easy to think about, really, is the baroreceptors are monitoring for stretch. And as stretch stimulates these baroreceptors, the signal is going to just pass through the typical sensory neuron. Okay, good old unipolar sensory neuron entering the spinal cord. And within the spinal cord, we're going to have a reflex, an automatic, involuntary, innate reflex that's going to act on the internal anal sphincter, parasympathetic motor neurons. It's also going to act briefly on the skeletal muscles, part of the reflex. We'll come back to that, though, in just a second. Let's talk about its actions primarily on the smooth muscle, though. This is the uh-oh moment. when you know you gotta go. The reason why is because this reflex causes the internal anal sphincter to relax. And it's going to cause the rectum wall, which is smooth muscle, to contract. So the internal anal sphincter is relaxing and the rectum is contracting, trying to push the feces out. Does that make sense? Again, I call this the uh-oh moment. Uh-oh. Good news. We have voluntary muscles that prevent us from just shatting our pants. That's good news. We get cerebral cortex, premotor cortex stuff. We can send a voluntary signal down to the external anal sphincter because as a part of the reflex, the parasympathetic motor neurons hijacked those skeletal muscles just briefly, and they actually cause the skeletal muscles to contract even tighter. So during the uh-oh moment, when the internal anal sphincter is relaxing, the external anal sphincter is actually tightening. And now you would need to make a conscious decision to relax that external anal sphincter. Cool? Everybody on board? So medically speaking, why is this so important? I think the easiest way to understand the importance of this is to think about babies and to think about geriatric. It is good to poop. We'll come back to that in just a second, though. Babies and geriatrics. Okay, babies are freaking all involuntary, y'all. In case you didn't know that, everything they do is involuntary. Like when you say your baby's smiling, I'm sorry, you know, I'm gonna smile and nod along with you, but your baby's probably not smiling at a young age. It's, you know, it's doing involuntary motions. Sometimes it's mimicking your face, which is pretty cool. But one of the involuntary functions is all about just pooping. That's what they do. They eat, they poop, they sleep. It's pretty cool. Why do they just poop their diapers? Hopefully they're wearing diapers. Because do babies have any control over their voluntary muscles yet? Nope. And when they do finally get control of their skeletal muscles, well, then you got to train them, you know, that pooping in a toilet's better than pooping in a diaper. And then it comes to a point where they just don't want to poop in the toilet because they're too busy playing. So they just, you know, kids are crazy. Next, let's talk about geriatrics. What starts happening to skeletal muscle in geriatric patients? Do they get stronger? No. Muscles, as we age, do something called atrophy. Atrophy, skeletal muscle atrophy, is when the muscle fiber starts getting replaced with collagen. So the older we are, each one of our muscle fibers, these big, huge, fused muscle cells, they die. And when they die, we don't make new ones. They just get replaced by collagen. So can't contract and relax collagen. There's a problem. Yeah, sarcopenia. It's one of the many issues. So in geriatrics, as we start to atrophy, it's not just our arms and our legs that atrophy, it's also all skeletal muscles, which include the external anal sphincter. And once that happens, we lose the strength, the power, and therefore in geriatrics, we also need to worry about involuntary fecal movements. Does that make sense? Cool. That's what I'm trying to get you guys to see, is that in the medical world, there's a lot of importance. Now, that could just be due to involuntary muscle spasms or, you know, just diarrhea causing, you know, liquid. The fecal matter is so liquid that you just can't hold that sphincter shut anymore. There's all sorts of issues that can come about down here with the defecation reflex. Okay. Okay. Awesome. So we're at the end of the road. We went all the way from mouth all the way to the anus. Let's look at what we got. Good old drag and drops. I'm hoping this is going to go fast and easy for y'all. So if I got a drag and drop from mouth to anus, we're just doing a flow chart basically. So the very first portion of this is going to be at the very beginning of your flow chart, which is the oral cavity. And if I follow this oral cavity through, I would be back at the oropharynx, the laryngopharynx, down into the, what is this structure right here? That's that esophagus. The esophagus has got an upper esophageal sphincter, a lower esophageal sphincter, and it all empties into the stomach. And we'll be learning about the different portions of the stomach down here. Okay, but the stomach has this enlarged upper region called the fundus. You got the body, you got the pylorus, you got the lesser curvature, and the greater curvature over the top. So we'll come back to all of that. Let's keep following the GI tract though. The stomach empties into what portion of our small intestines? What's the first and most important portion? Yeah, not the cecum yet. That's the first portion of your large intestines. This is going to be the duodenum. Hopefully everybody's following along. Slow me down if you need me to slow you down. Now, from here, it's going to go into the other small intestines. And I don't see jejunum on here. I'm going to go ahead and add a jejunum, because I could test you on this for the jejunum. Now, the jejunum, basically, if I put an arrow to kind of this upper portion right here, right there. Not way down at the end of the road, not the duodenum, just somewhere up here, okay? This all is jejunum up here. You guys would answer that as jejunum. Is that clear? And now, if I just put an arrow way down at the end of the road, somewhere down here, okay, anywhere down here, that would be the end of the small intestines, which would be the ileum. Everybody feel pretty comfortable with that? Now, the ileum has an ileocecal valve. So from the ileum, that food is that chyme is going to pass into what? What is this chamber down here? That's the cecum. What is the little tail on the bottom of the cecum called? That's the appendix down there. Now, from the cecum, we're going to go up towards the liver. So this portion of that cecum now changes into a colon, but it's going up. Good. So that's the ascending colon. This is going to pass across. That's your transverse colon. There's no arrow to the descending colon. That's kind of funny. It's going to pass down from the spleen. This big purple blob right here is the spleen. So that is going to, gosh, there's no arrow going to the spleen either. That's kind of funny. That's the spleen right there. But anyways, descending colon, then you're going to get this big S turn, which you can't really see very well on this particular view. If you flip it over, you're going to be able to see the sigmoidal colon a lot better. Good, sigmoidal colon a lot better. But all that's going to pass down into the storage container right here. What's the storage container going to be? That's the rectum. And then the opening down here is called the anal canal, and that all is going to be able to pass through the anus. The last of these, this funny little yellow tongue, that's your pancreas. That's going to empty material into the duodenum. The gallbladder is the green ball on the liver, and the liver itself obviously is this big dark brown blob. Everybody feel pretty comfortable? Love it. I don't know, Ashley. I might have misspelled it earlier. Maybe that's what the problem is. I'm sorry if that's true. Teach yourself by placing the structures listed in the order in which they are encountered by your chips and queso. So you're just going to switch these guys around, all right? God, I want some chips and queso now. But you're just going to switch these guys around. There's no arrows. You're just thinking pure on. Oh, yeah, Kirby Lane's got some of the best chips and quesos. Of course, I think Torchy's is probably my favorite. But, yeah. Taco. Whatever. What's the name of that taco? Whatever. Anyways, can you guys work on doing this when you guys work on doing this? You're just going to say, what's the first thing this is going to come into contact with? Let's see, lingual frenulum. I haven't really taught you guys about frenulum yet. I will in just a minute. Lingual tells us tongue. So this looks like it's the uppermost. Then from there to the back of the throat where we find the uvula. So you'll just kind of have to take and restructure these. Gastroesophageal sphincter is going to take us down into the stomach. Inside the stomach, we're going to come across the verruga. So anyways, you guys can kind of work on this. What is the plicae circularis? Great question. Actually, it's something I should have covered. Well, that's something I will cover. That's what's going on. Just haven't covered it yet. It's found in the duodenum and the rest of the small intestines. So if we look at the duodenum and the rest of the small intestines, you guys see these big, gigantic waves? They look a little bit like ruga, but they're not ruga. Okay, these are not elastic waves. These are, they're just gigantic folds in the tissue, and they're covered in villi, and the villi are covered in microvilli. And I have a slide for it all somewhere. I just don't remember where the heck it is. But what that ends up doing is it massively increases the surface area. Massively increases the surface area of your small intestine. So what's that going to do to the amount of material that can be absorbed? I was going to get to this in a bit anyways. So the greater the surface area, the more absorption can occur. Yes, no, maybe so? Okay. Well, I guess while I'm here talking about this, has anybody heard of celiac disease at this point? I mean, it's pretty common nowadays in terms of conversation about celiac disease. So celiac disease is where we take those plicae circularis. Okay, the normal plicae circularis is covered with these big finger-like projections called villi. And in celiac disease, the villi go away. Okay, they get destroyed. You actually have an autoimmune function that comes and starts to chew your villi away. So what just happened to the surface area that absorption can occur across? Increase, decrease, no change. And we get a huge issue with the absorption of nutrients in a celiac disease patient. They just don't get the nutrients that they need anymore, whether that's glucose, fatty acids, vitamins, calcium, it doesn't matter. All absorption decreases in celiac disease. This cool image. The nutrients are bouncing off. Yeah, it's pretty nasty. You have to really control what you're eating, smaller portions in your meals, super painful. There's issues. So that's what celiac disease is it is basically these plicae circularis of these small intestines, these big villi. Okay, this is the ileum. Look, I love this. Here's the duodenum, here's the jejunum, and here's the ileum. Which one has the greatest function in absorption of materials? Which one has the greatest surface area? I mean, it's all about the surface area, right? I mean, it's the more surface area there is. So these big, huge folds, these are the plicae circularis. Sound good? Cool. All right, stomach anatomy. Stomach anatomy, I mean, just work your way with a flow chart. That's always my recommendation. Always my recommendation. If I'm taking and I'm putting food in my mouth and it's going down through the esophagus, in order to enter the stomach, what's it gonna have to pass through? Cardiac sphincter, which is also known as the gastroesophageal sphincter, because of course nothing could be simple, but this is the lower esophageal sphincter. Oh, there's always multiple names. Sorry. You guys don't need to know it as gastroesophageal, but that should make sense. Gastro, stomach, esophageal, sphincter. So that's the sphincter. I'm going to be able to show the sphincter from the inside. From the outside cut, I can't really do that very well. The region, so if I say, please pay attention to my wording here, what region is this? This region of the stomach right here is called the cardia. The cardiac sphincter is associated with that region. So if I say, what region is this? You'll say cardia. If I say, what structure is this? You'll say cardiac sphincter or lower esophageal sphincter or gastroesophageal sphincter. Does that make sense? Please read those lab practical questions because some of them have 2 questions. Name the structure, name the organ, name the structure, name the region. You have to read the questions and make sure that you're answering them accordingly. The next region is this upper region of the stomach. Does anybody remember what I've been calling this area? Apparently it's where all the excitement happens. It's where all the fun happens. That's your fundus. The large majority of your stomach is just body. So all of this is the body. And then this white portion down here, that's the pyloric region. Make sense? Based on everything we've learned so far? If we look on the inside, the pyloric region would be here. So the end of the pyloric region, there is smooth muscle, round, circular, there's a sphincter. That's going to be the pyloric sphincter. Yes, no, maybe so? What are all the folds inside of your stomach called? Well, you can call them gastric folds or gastric rugae. Please be listening to my words there, gastric rugae. You're going to see that there's other rugae. There's urinary bladder rugae. There's vaginal rugae. There's rugae all over the body. Rugae are just elastic folds that allow for expansibility of those organs. Yeah, sure. Yeah, if that'll help you. I mean, yeah, for sure. That's something you guys can do too. Remember, you guys get this drag and drop. So, and then that's not greater. Greater curvature is the larger of the curvatures, and lesser curvature is the smaller of the curvatures. Now, the importance of those curvatures can't really be understated. They are important. I've saved them for last because I want to go to this picture right here, and you'll notice that there's the stomach right there. And there is the greater curvature of the stomach. And do you guys now see that there's this gigantic white sheet hanging down from that greater curvature? It's actually attached to the greater curvature. This big, huge white sheet, this is fat. Okay, yeah, it's like a blanket. I think of it like a curtain. I like to think of this like a curtain. This is called the greater omentum. This is what gets bigger when somebody gets a beer gut. Okay, the beer gut is the greater omentum building up carbohydrate storage. Well, carbs aren't stored as carbs, they're stored as triglycerides. The body takes and converts all those carbs into triglycerides and stores them away inside of adipose tissue. So this greater omentum is that. No. Oh, I don't know. I thought I grabbed stomach, but it must have been right next to it. Sorry about that. Yeah, that's the stomach I was using. Anyways, thank you for pointing that out. So this greater omentum is like a curtain. What that means is that curtains are attached at the top. Curtains are not attached on the side, and curtains are not attached on the bottom. Does that make sense? The greater omentum is really cool during surgery because what you'll have to do is you literally, I wonder if, yeah, there it is. God, that's so cool. You literally lift that greater omentum up and away, and it looks like this crazy sheet of, I don't know, fatty tissue. This is tripe. For anybody that eats tripe or know what tripe is, it's this huge sheet of fat that hangs over, yeah, menudo. It's this huge sheet of fat that hangs down from the greater omentum all over that abdominal cavity. And you cannot do an abdominal surgery without thinking about the greater omentum. You're going to lift it up like a curtain. You're going to hang it off to the side. You do your abdominal work. You put the greater omentum back in place. It's super cool. It's so cool looking. You lift it up, then you can get to the intestines. Then when you're done, you put it back. During a C-section, they're actually doing their incision below the greater omentum. They're down in the pelvic cavity, not the abdominal cavity. So the incision for the C-section is actually way down here. So they're going inferior to that greater omentum. Clearly. All right, everybody feel like they could identify the greater omentum? Now the lesser curvature, I don't have a really good picture for it, but the lesser curvature has another sheet of mesentery that connects it to the liver. So the liver is up above it and that lesser curvature attaches to it, but I won't test you on any of that part. I just want you guys to be aware the stomach's got to be held in place. Otherwise the stomach can flip and do all sorts of crazy stuff. Make sense? All right, yeah, so in this one, we can label the transverse colon. We got a little piece of bladder sticking out. Bladder's always going to be green because of its function with bile, and bile is green. Liver's this gigantic brown gross thing here. Always the bridesmaid. Ileum, end of the road. So where the ileum comes into contact with the cecum, that's your indicator that it's the ileum, okay? Always the ileum right there, the last little portion of the small intestines before it becomes that cecum, and then coming up from that cecum is the ascending colon. Makes it to the liver, flexes over, 90-degree angle, transverses across, makes it to the spleen, 90-degree angle, flexes down. Yes, no, maybe so? Let's see here. What can we do that's different? Now, in this particular model, the liver has been taken out, the stomach's been taken out. So what you can see down below all of that finally is the pancreas. Everybody feel pretty comfortable that this organ is the pancreas? It's attached to the spleen on one side, and then on the other side, the pancreas is going to be creating a bunch of digestive juices. Yeah, it kind of does look like a tongue for sure. I keep using that analogy too. But this is a duct that's going to secrete all of its digestive juices into what portion of your GI tract? Where does the pancreas secrete its goodies? I'm nodding, Andy. I'm just waiting for more answers. This is the duodenum. If you remember the flow chart we did, I guess yesterday, I mentioned that the liver, gallbladder, pancreas, all of these juices go back into the duodenum, okay? The duodenum does the large majority of the work. Yes, no, maybe so? So pancreas, this little piece right here is the duodenum. Also, if you recall, this thing back here is retroperitoneal. So it's surrounded by adventitia. It's actually behind all of the parietal peritoneum. And the large majority of the peritoneum, well, that's all that, oops, sorry about that. That's all of this crazy mesentery stuff. So this sheet of connective tissue that holds the transverse colon to the pancreas. Does anybody remember what I called this just a little bit ago? I guess right before our break, I mentioned this thing. So this is the transverse colon. That right above it's going to be the transverse mesocolon. We just throw the word meso on it and it becomes mesenteric. So anything that's meso is mesentery. Make sense? The big sheet of fat, once again, greater omentum. It's hanging off of the stomach, but the stomach's been removed. I got apparently two, oh yeah, I do have two pieces of esophagus. Because the esophagus, when I remove the heart, this big muscular tube behind the heart is the esophagus. And that esophagus descends, passes through the diaphragm. And where it passes through the diaphragm, that is also the esophagus right there. And that's right where it's going to connect to the stomach. Passes through into the abdominal cavity, boom. I don't really think I need to go through ascending colon again. More of the small intestines. This is a difficult area because it depends on the patient and how their small intestines wraps. This could either be the jejunum or the ileum. It just depends. Definitely not the duodenum though. And finally, last but not least, this gray white line that holds your large intestines together, Taenia coli. Make sense? We're just zooming in down here at the junction between the small intestines and the large intestines here. That's it. Shouldn't be too crazy. This opening to the large intestines, that's your cecum. The valve, these two white lines are part of the valve. These are the membranous flaps that go from the ileum into the cecum. So that right there, if I were to point anything on a lab practical to that right there, that's the ileocecal valve. Make sense? Ileocecal sphincter, it's not really a muscular sphincter. We can control the valve opening and closing just a little bit. There's hormones that can actually do that, but yeah. And then just like the rest of the small intestines, there's plicae circularis or circular folds. All right, I'm not gonna take too much time labeling all of this. Most of this we've seen before. But what I want to get you guys to see are a few new terms and then a couple of the old terms that I started teaching you guys in the lecture portion. So each enlarged region is called a haustra. This is one students miss on the lab practical all the time because I will circle like this right here on a lab practical. I'll just circle this area. I'll turn it. I'll make that into a dotted line. And no, I need an outline. I need an outline. I need no fill, that's what I need. Right, so I'll circle that right there and I'll say, what is the structure circled? And people will say, that's transverse colon. No, it's not. Transverse colon is this whole thing. Okay, if I said what region of the large intestines is this, then you'd say transverse colon. But this structure right here is a single haustrum. Does that make sense? Cool. I've mentioned this a couple of times, but down here we have the cecum, and that cecum is going to ascend. It's going to keep going up, keep going up, keep going up until it makes it to the liver, at which point it's going to take a 90-degree turn and become the transverse colon. This point where it turns is a critical point. Patients have issues sometimes with this turn, and it changes how well they can digest. But these are called flexures. There's two terms for it. One, it has to do with just anatomical position. So is this the patient's right side of the body or left side of the body? So we can just call it a right colic flexure. But another term for it, and the term that's on my lab objectives, is hepatic. Because where is it turning? It's turning when it makes it to the liver, and liver in Latin is all about hepatic. Make sense? So now we're going to go over to the left side of that body, just at the just inferior to the spleen. So that area right there, that area right there, okay? That's going to be the left colic flexure, but it's also just known as the splenic. Oops, yeah, that works. I'll keep it consistent with the other one. Splenic flexure. Make sense? Oddly enough, not everybody's spleen is on the left side of the body. 99.9% of people's spleens on the left side of the body, though, but sometimes people have bizarre spleens. Sigmoid, good old S turn, right? Sigmoid. As we descend, we go into this S turn. And yet again, we don't have a label for that portion. This is all descending colon over here, descend, descend, and then it makes this big S turn, sigmoid, and that goes into that rectum. Last but not least, that white line is the Tenier coli. Are there any questions on that one? Do you guys feel pretty comfortable with these lab objectives? Okay. Next, we're going to get some mid-sagittal cuts now. Okay, these are actually repro models, but repro models are perfect for looking at the end of your digestive system, the end of your GI tract. So for instance, yep, good, I'm glad. A lot of people don't get to see that uterus, or maybe think that's the bladder. That's not the bladder, that's the uterus. But anywho, so just posterior and actually just superior to the uterus right here, this is the very last portion of the colon. Now, this colon is S-ing around, right? And it's going to descend down into the rectum. The rectum's a relatively large chamber covered in baroreceptors. Those are just, this area is just a storage container for fecal matter. Sorry, I was a little confused by what Tariq said there for a second. The canal, the open space here, that's the anal canal, and then the end to end of the road, that is the anus. So GI tract runs from mouth to the anus. You do have blood vessels, lots of blood vessels throughout the entire area. I'll actually, yeah, I'll talk about the anal canal in just a second. It's not a bad thing to think about. Yeah, I don't know the reference between toxins and rectum, Heather. So that might be something you can teach me. If there's some toxins you know about that cause like rectal spasming, is that what you're referring to? I don't know. Oh, yeah, there's a lot going on there. If you're talking about buildup of toxins, yeah, okay. There's a lot going on there. I don't have the time to talk about it now, and I don't have the expertise to really condense it and shorten it. That takes actually a lot of thinking and whatever understanding of the rest of the system to really understand that. In terms of Man, if constipation occurs for like 3, 4, 5 days, you can definitely get, forms of inflammation and sepsis and bacteria buildup, and it can cause a lot of downstream issues later in life too, damage to the entire rectal cavity. So we'll come back to hemorrhoids. That's one that I do know a little bit more about, so we'll come back to that in just a second. But let's finish this up. We got anal sphincters. Which one would I be pointing to here, inner or outer? Students get this wrong every semester, so it is what it is. Okay, this is internal. It's internal. It's closest to the anal canal, and then this one's external. Now, which one of them is smooth muscle? Internal or external? Which one is smooth? Good. And then which one would be skeletal then? It's going to be the external. So let's go back to lecture real quick. This one. And let's go to this, end of the road, rectum, rectal valve, that sort of stuff. Now, you guys don't need to know the levator ani muscles, the rectal valves, hemorrhoidal veins. That's interesting, right? Might have something to do with hemorrhoids, but the reality is that these anal columns, so this is the anal canal right here. This whole area right here is the anal canal. And inside of the anal canal, you guys see these raised lines. They're called anal columns. And at the very end of these anal columns, well, there's anal valves in between them, but at the very end of these anal columns, there's like a small opening, a small hole. And what ends up coming out of here is a mucus-like secretion that's filtered from your blood that helps with defecation. It helps to lubricate the anus during defecation, and definitely, makes it a smoother ride, so to speak. Now, what happens in people is if these anal columns get blocked over time, then basically you end up with inflammation of the anal columns and inflammation around that anal canal. That is hemorrhoids. Okay, it's inflammation of these anal columns, and it makes, defecation extremely painful because the anal columns aren't producing the proper amounts of mucus and moisture anymore. So every single fecal movement's going past inflamed and pained tissue that doesn't even have extra mucus to help protect it. Fun times, right? Occurs from people that sit too much, like truck drivers and stuff like that. If you put too much pressure down here, or people that sit on the toilet for like 30 minutes at a time, maybe because they're trying to get away from their family and playing video games on their phone, that sort of pressure could end up causing hemorrhoids as well. All right. Let's take a short break. What's going to happen when you guys get back? Twofold. We're going to finish up the anatomy of the accessory organs. So what we're going to talk about is the liver and how the liver plays a role in digestion. We won't talk about the physiology of that yet. We'll bring into play a little bit of bile, which is some physiology, but we're, again, not going to talk all the way about what's happening with digestion and absorption. We'll leave that for tomorrow. So we're going to have a little bit more of the anatomy of gallbladder and how bile flows down into the duodenum. And then we're going to talk a little bit more about the pancreas. We're going to tie this in with your lab objectives. Because for your lab, you need to be able to identify the pancreas, and then we're going to go into some histology, and we're going to be identifying the cells of the pancreas. So again, we're going to tie in some basic functions, but we're not going to talk all about the function of the pancreas. Not today. We'll leave that for tomorrow. That's going to be what we do for the rest of the day. What does that mean? If you look at the schedule, it's just not going to work. I wanted to fit in urinary anatomy here to make the next unit a little bit easier. That ain't happening. So urinary anatomy, if you pull out your schedule, please move this down. We'll do urinary anatomy July 12th-ish. Urinalysis lab is crazy short. Urinalysis lab is literally a 15-minute lab. It's so short. We're just learning what the importance of urinalysis is, how you'd go about measuring urinalysis, and then I'm going to give you guys like, a PowerPoint that you can walk through. So we will be using this time to go through urinary anatomy. What does that mean? Do you guys have urinary anatomy on your lab practical this Sunday? Yes or no? No. Okay, we just didn't make it, but we will cover it in detail. We just are going to wait until July 12th for that. So there will not be a study guide for urinary anatomy due. So the only study guide that's going to be due and actually this is not going to be Sunday, this is going to be Friday as we've been keeping consistent on. But the digestive, the one that we're going through right now, the digestive anatomy study guide will be due this Friday at midnight. Cool? I don't want to test you guys on things that we haven't covered in class. That's not my thing. I'm not going to just say, okay, we didn't cover it, therefore the work is on you. It's just not fair. You guys got plenty of stuff to go through. All right, cool. So let's take a break. Take it until 30 after. I will try not to go too much past 1 today. When you guys come back, we'll just have a 30-minute little session. I know you need a break now, you're tired. And in this session, we'll talk about liver, the gallbladder, and the pancreas. Okay, go, 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 go. 30 after. We'll probably go a couple minutes past 1, Melissa, but not too much past 1. So if you got to go, then you'll just go, and we'll try to make it as concise as possible. All right, let's get this show on the road. We'll finish it off here. Everybody ready? All right. So what we need to do here is we need to somehow relate the liver and the pancreas with the rest of that GI tract, and we've kind of already done this. Where this enters the system is all related to the duodenum. The duodenum is all about digestion and beginning that absorption process. The rest of the absorption will occur down here in the jejunum, but the large majority of digestion occurs in the duodenum. The pancreas produces something called digestive juices, pancreatic juices. The liver produces bile, B-I-L-E, bile. Each one of these is important in digestion. How? I've been referring to this a few times, but the reality is that when we're talking about digestion and the digestion system, what we really need to be thinking about are the nutrients that we're getting out of our food. And the way to think about these nutrients, these macronutrients, is biomolecules, is that they're essential for all cellular function. And they're pretty darn easy to think about because every single time you've looked at the back, the nutrition label, buy a bag of chips or crackers or whatever the case may be, you look at that nutrition label, and what are you guys seeing on that nutrition label? What's one of the first things that you guys are looking at? Calories, exactly. What exactly is a calorie? A calorie is just the energy. It literally has to do with joules. It has to do with the energy that's available in the food we eat. But that's not a macro molecule. That's not a biomolecule. Help me out. Ah, there we go. Carbs. So one of the first things everybody looks at is carbohydrates, for sure. Carbohydrates. And what we started to notice with carbohydrates is we begin the digestion process of carbs. We begin that in the mouth. Next, what's happening? Can somebody else give me something else on the nutrition label? What else are we digesting? Sugar is carbs, so proteins, awesome. And what else? Fats. These three things are found on that nutrition list for a reason, because these three things help build your cells. The last thing is nucleic acids. These are the four major biomolecules. And this is what your digestive system is all designed for, okay? It's designed to digest these biomolecules, these big macromolecules that the rest of your body is made from. Carbohydrates are big long strands of monosaccharides. Proteins are strands of amino acids. Fats, different types of fats like steroids, fatty acids, triglycerides, so forth, so on. Nucleic acids are important for DNA and RNA. And we need to get this stuff out of the food we eat. How do we do that? We use different digestive tools, okay? For instance, salivary amylase helps us digest carbohydrates. Does that make sense? Proteins, we've already seen. Pepsinogen gets cleaved into pepsin, and pepsin helps to digest those proteins. Does fake saliva? I'm not sure what you're referring to, Melissa. I've never heard that terminology, fake saliva, before. It doesn't mean that I don't know everything. Who don't salivate anymore? No. So is fake saliva that you give to a patient that can't salivate anymore? So I haven't heard of this. If that's what's going on. Oh, cool. I would assume that they probably add some amylase to it. Yeah, I don't know. I've never heard of it. There's all sorts of stuff in medicine that I haven't heard of before, so I'll have to look that up. Okie dokie. We kind of have this idea going on right here. Yes, no, maybe so. We're going to be walking through the different digestive enzymes that do each one of these. You guys have already kind of learned the beginning process of some of these. For instance, fats, lipases that are produced in salivary amylase or in saliva. These are activated down in the stomach, and we start digesting fats. Now, the liver. How does the liver play a role in this? The liver produces bile, and it's not just bile. It's actually a bile salt. Bile salts are important in the digestion of fats. Most fats cannot be digested without bile salts. Okay, bile salts, we're going to go into the physiology of bile salts probably tomorrow, but bile salts are going to be made up of, well, a composition of primarily cholesterol. Okay, bile salts is primarily a cholesterol. So what do we know about cholesterol? Polar, non-polar? Non-polar. Cool. And then the other side is going to be a salt, hence salt, right? So it's going to be something like a sodium, typically. Salts have charges. So one side is not charged, it's non-polar, and the other side is charged. This sounds a little bit like surfactant or a phospholipid, doesn't it? One side's going to be attracted to water. This side over here is water soluble. And then this side over here, and the cholesterol, is lipid soluble. So one side is going to be able to bind up fats, and the other side is going to be able to be attracted to water. This helps us with a process called emulsification. Bile salts help us emulsify fats. The emulsification of fats, imagine a vinaigrette. Yeah, cell membrane, phospholipids, that sort of stuff, anything can interact here. But imagine a vinaigrette, right? You got oil and you basically got water, you got vinegar. Do oil and water mix? Nah, right? They're not going to ever mix. So what we need to do is we need to figure out a way to make those fats, that oil, mix with the water. And the way we do that is with salts. And that's where the whole vinegar salt comes into play, is that it's going to allow for, when you shake up that vinaigrette, the fat, it doesn't dissolve in the water. It's just not possible. But when you shake up a vinaigrette, that oil mixes better with the water. And then over time, it's still going to leave that solution. It's still going to separate out into an oil and then the rest. You guys know what I'm talking about? Kind of? If not, you can look that part up. That's called emulsification. So this bile is essential for the proper digestion of fats, because we cannot digest fats unless we emulsify them first. After we've emulsified fats, other enzymes can now digest those fats. We can't properly digest fats until we emulsify them. Okay? That's where the liver comes into play and those bile salts come into play. Now, where do we store those bile salts? The liver makes these bile salts, and it's going to send those bile salts into the gallbladder. And now those the gallbladder is going to end up helping to secrete all of that bile when we need it into the duodenum. We're going to draw another flow chart here soon. I'm just trying to get you guys to see that fats simply have a very hard time getting digested without this emulsification process occurring first. It's almost impossible. All right, now what about the pancreas? The pancreas secretes, its primary job is not insulin and glucagon, by the way. Okay, that's something we saw in Unit 1, is that the pancreas have the islets of Langerhans, and those secrete glucagon and insulin. But the primary role of the pancreas is to secrete digestive juices. Now, digestive juices are made up of 4 things. 1, pancreatic amylase. Well, what do we know amylases help us digest? Not fats, the fats are lipase. So think about the salivary amylases. They're all about the carbohydrates. More importantly, starches. Okay, so pancreatic amylases are digesting starches. Okay, starches are big, long chains of glucose bound to glucose, bound to glucose, bound to glucose. Yes, no, maybe so? Two, pancreatic. We'll go peptidases next. Peptidase. There's actually a number of different peptidases that come out. Pancreatic peptidase. So what are we going to be enzymatically cutting down with peptidases? Proteins. 3, pancreatic lipases. So what are we breaking down with these different pancreatic lipases? Fats. Now again, lipases can't really digest these fats unless they've been emulsified first. Just make sure you understand that. And then 4, you guys starting to see what I'm doing here? Amylases for carbs, peptidases for proteins, lipases for fats. So what's the last one going to be? Pancreatic nucleases. So this helps us digest nucleic acids. Make sense? Sure sounds like the pancreas has a pretty critical role in digestion. Okay, in fact, the duodenum, the small intestines, they cannot digest on their own. They simply can't. They require pancreatic digestive juices. They must have it. The pancreas goes wrong, everything goes wrong. So this is an intro to why the anatomy of this right here is so darn important. Because if the liver's producing these bile salts, correct, Tariq, if the liver is producing these bile salts, I mean, I guess the answer is yes, Tariq, because we can actually put in digested nutrients into our patients. Just not a great way to, quality of life kind of thing. All right, but the liver is producing the bile salts, and we're going to need to get those bile salts from the liver down into the duodenum. The pancreas is producing digestive juices, so we need to get those digestive juices from the pancreas into the duodenum. Everybody with me? One last thing to think about is that all of the material that's entering the duodenum, this chyme, is coming from what organ? What's just upstream from the duodenum? What's upstream from the duodenum, everybody? The stomach. And what is the stomach producing? Secreting 3 liters of a day. Hydrochloric acid. That hydrochloric acid is leaving the stomach and entering the duodenum. Gulp. What's this duodenum going to have to do with all of that hydrochloric acid? Just take it? Toughen up? Suck it up? No, it's going to have to neutralize it. Does that make sense? Now, the small intestines produces a little bit of bicarb, but the rest of the bicarb's coming via bile and pancreatic juices. So another main composition of both of these structures, okay, the pancreas is also going to be secreting lots of bile. I said bile, sorry. No. Bicarb. The pancreas is also producing lots and lots of bicarb. It's going to be producing sodium bicarbonate. And our bile salts, the last component that I really care for you guys to know, bile salts also have bicarbs in them. Because the neutralization of the acids is a pretty high importance. Does that make sense? Cool. So let's get down here, 45 after. We're doing good. We're doing good. We're doing good. So the liver, let's go there first. The liver is producing, where's that picture? Do I have it here? Lo siento. 1 second. I have it somewhere. I saw it earlier today already. I just got to remember where I have it. No. Yes, there it is. I'm sorry about that. So the liver. The liver is a huge organ that has many functions. Always the bridesmaid, never the bride. One of its major functions, I mentioned this earlier this semester, is to detoxify your blood. And if you recall, I showed you guys some websites that talked about the hepatic portal system. Do you guys remember this when we were talking about blood supply? We were talking about the hepatic portal system, hepatic portal arteries, hepatic portal veins, that sort of stuff. And I was referring to the fact that all of the nutrients that we get from our small intestines and large intestines, all the ions and water, everything's got to go back up through the liver, and it's going up through the hepatic portal system. Yes, no, maybe so? So when you take your liver and you start breaking your liver down, you start looking at the different portions of the liver, there are lobes of the liver, multiple different lobes, caudate lobe, quadrate lobe, left lobe, and right lobe. Those are the 4 lobes. Caudate lobe, oh goodness, they're not individual. Caudate lobe right here. This is up by the inferior vena cava. The left lobe is all left of this kind of ligament line that you guys see here. The quadrate lobe, it's a really nice square. 1, 2, 3, 4. It's got 4 distinct corners. Quadrate, quad, 4. And then the right lobe is the largest of the lobes. It basically almost the entire right side of your liver. If you look at it from this view, all you can see is the right lobe, which is the biggest portion, and the left lobe. You can't not see the caudate or the quadrate. Yes, no, maybe so? Now, if you start to take those lobes and you start to cut into those lobes, what you're going to see is inside those lobes is a whole bunch of lobules, right? Lobes, what's smaller than a lobe? A lobule. Does that make sense? So if I start to cut into those lobes, this right here with the purple border, This is one lobule. Here's another lobule. Here's another lobule. Here's another lobule, so forth, so on, okay? Each one of those is a lobule. The whole thing is a lobe. Each one of these is a lobule. And if I zoom in on this one, this is a good one right here to zoom in on. Do you guys notice that there's a circle right in the middle? That's the central vein. Oh yeah, that's the one that it chose too. That's a good one right there. This is a central vein, and veins are defined as veins because they return to the heart. Make sense? So the central vein is going to be feeding all of the blood back to the heart. So I'm just kind of bouncing back around between different pictures because I think each one helps. So all of the hepatic portal system is going to enter the lobes and it's going to enter the lobules. Then the liver is going to filter all of that. And as it filters it, it's going to end up returning that blood to the central vein. And then that central vein is all going to go into the hepatic veins, and all those hepatic veins end up feeding back into the inferior vena cava before returning to the heart. Now we can see that with this picture right here. This is a great, just drawing of this. This is the central vein, and this is 1/4 of a lobule. So if we go back to our histo picture, right? This is one lobule, and if I were to take 1/4 of this, that's what I'd be looking at. Does that make sense? One big piece of the pie. These are hepatocytes. They tend to be, sorry, these aren't hepatocytes. This is the liver lobule. They tend to be relatively like, I don't know, hexagonal in shape, or, 6 sides, 8 sides. It kind of just depends on the lobule you're looking at. All the models are going to be represented as basically 6 corners. Every single one of the models is going to basically have 6 corners. And in the corner of each lobule, so let's go back to this picture here again. In the corner of each one of these lobules, do you notice that there is a grouping of vessels? Grouping of vessels there, grouping of vessels there, grouping of vessels, grouping of vessels, and this kind of gets cut away, but there's a grouping of vessels there. These are known as portal triads. Okay, so portal triads right here. The portal triads are basically a cluster of different tubes. They're not all blood vessels. They're a cluster of different tubes that are going to be feeding material into the lobule. The large majority of the lobule is made up of hepatocytes. So if I point to any of the cells here, say for instance in this model, or in, I have another model for you guys. One second. It's over here. Digestive liver and pancreas models. So there's another drag and drop for liver and pancreas here. So if we go down to this model right here, this is what could be on your lab practical. What you'll notice is this corner over here is that central vein. This is a larger view of it. So again, this is a zoom in on 1/4 of one lobule, but the entire lobule is indicated by this kind of columnar structure right here. So right there in the middle of that, that's the central vein. Does that make sense? That would be the central vein. And then I'm taking a microscopic cut to be able to look at this structure over here. So this is one lobule. Did that help, Melissa? You guys see? Everybody see? Now in the corner, there's one there, there's one there, there'd be one where one lobule meets another lobule, so there'd be another one right there. That's where all these blood vessels come together. Okay, these are called a triad. This is the portal triad right here. All of this together is going to be the portal triad. I'm not really a big fan. I guess it has to do with the way the words are going to be in. But all of these tubes together are all called a portal triad. The big one And the easiest one, I hope you guys can remember because this is something you guys have learned about before. This is the hepatic portal vein. This is bringing back nutrient-rich food from the small intestines and large intestines. This is bringing back water. This is bringing back vitamins. This is bringing back toxins. All the good things and all the bad things. Everything's coming back through the liver through the hepatic portal veins. These are pretty small because they're going out to the thousands upon thousands of lobules that we have at this point. Yes, no, maybe so? And what you'll notice is that there are these cracks now that lead away from the hepatic portal vein, leading into the lobule. There's all these cracks, tons and tons of cracks. And you'll notice if you follow the direction of the flow, it's going from the hepatic portal vein and it's flowing towards the central vein. Yes, no, maybe so? All of these cracks are all part of the liver sinusoid system. They're all liver sinusoids. So there'd be a liver sinusoid there, sinusoid, so forth, so on. Okay? Not just one sinusoid, there's multiple sinusoids. Yes, no, maybe so? Awesome. Now, one more thing before I get into the nitty-gritty of going inside is that all cells of your body require oxygen-rich blood too. Well, that's going to be coming in through an artery. So the hepatic, what just happened? So the hepatic artery is the red artery. It's part of the triad. Triad means three. Branch of hepatic artery, branch of the hepatic portal vein, they all come in together. And you'll notice that this is going to feed oxygen-rich blood into those liver sinusoids as well. And that's going to go out into the capillary beds. And it's going to feed the cells. Each one of these, 1, 2, 3, 4, each one of these is a liver cell. Every one of these is a liver cell. Yes, no, maybe so? Oops. I was going to zoom in. And liver cells are hepatocytes. So if I pointed to this one, it's a hepatocyte. If I pointed to this one, it's a hepatocyte. So forth, so on, down the road. All right. We'll come back to one more thing I want to add to this. I'll end up making another thing for it. I'll make another thing for it. We'll deal with this one in just a second. All right, so the physiology of this goes as follows. We still have a few more things we have to talk about over here. Physiology of this goes as followed. We get this nutrient-rich food, toxin, water, whatever, coming back from the digestive system. We got oxygen-rich and nutrient-rich blood returning over here. All of this is going to be passing by the hepatocytes. And the hepatocytes do what they do. They take the proteins out of this nutrient-rich blood. They take the glucose out. And they pull all of it out and they start to process it. They make the proteins, right? They're making 90% of your blood proteins, for instance. Other amino acids are just going to send on to where they need to go. Glucose, they take and store glucose away. Oh, most of your blood's glucose, most of your body's glucose is stored in your liver as glycogen. What else is going on? They're going to take cholesterol. They're going to take salts, and they're going to start to take and convert those into bile. The hepatocytes are making the bile. Yes, no, maybe so? Dang, time goes by. Now, as they produce the bile, they're going to push the bile out of these tiny little green dots that you guys see here Each one of these hepatocytes is associated with a tiny little tube. You guys see these little green dots? Green dots up and down the board, green dots. And those green dots are all interconnected. These green dots are all going to be connected to this green line you guys see right here. Okay, these are canals, but they're very, very small canals. These are all going to be bile canaliculi. Okay, these are small canals that are all linked together. And I think I have a good image of how they're linked together right here. So if you look at this picture, you guys see hepatocyte, 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 hepatocyte. These are the bile canaliculi, and they're all interconnected. They're all woven together. Yes, no, maybe so? Perfect. Those bile canaliculi are feeding back. So all the blood's coming in and the bile's going out. So the last part of the portal triad, the last part of that portal triad is green. And green on your models is always going to be associated with bile. And these are the bile ducts that are leaving the liver to go enter the duodenum. Okay? Make sense? Paka! All right, that's a hard one for students. So make sure you walk back through that. You can watch back through it to be able to find it. Okay, so different lobes, you guys can label those lobes now. Inferior vena cava right there, inferior vena cava. We got purple is portal. Purple is portal. I love that little saying. But if you see on a model, you see a purple blood vessel. Purple is portal. Go back to this. You taking a screenshot? By the way, purple, right? Purple. Purple is portal. Anytime you see purple blood vessels, we're talking about the portal system. So purple here, hepatic portal system. Green, bile, red, arteries, blue, veins. Sweet. You guys can go through and label that. Now, bye, Melissa. Now, the last part that's really difficult for students is not the pancreas. Pancreas is pretty easy for students. Histology can be a little complicated, but I think you guys are gonna be able to do that just fine here. Let's go just real quick, do a flow chart where we start with these bile, actually let's start with hepatocytes. So we're gonna say our hepatocytes, you guys can actually have flow into this if you wanted, which would be the hepatic portal vein and the hepatic arteries, bringing blood to the hepatocytes. Now, one of the functions of the hepatocytes is to produce bile. So we're going to make that bile, and that bile is now going to move into the bile canaliculi. Gosh dang it. From the bile canaliculi, it's going to move into these tiny little bile ducts. Bile ducts. Now, we often will call these bile ductiles because they're smaller ducts. And if you go to the model here, we're now zoomed out on models like this. And I'm always going to test you guys on a model like this. The hepatocytes are out here in the liver lobes and the liver lobules. The bile ductiles are now going to be carrying that bile into these bigger tube systems. So the very first of those two tube systems, this green line, that green line right there, these are still in the liver. And as long as they're inside the liver, we're going to call them hepatic. I hope that terminology makes sense. One's coming from the right lobe, the other one's going to be coming from the left lobe, And so it's all about anatomical position. We got a right hepatic duct and we got a left hepatic duct. So these are arriving from all of these bile ductiles. So if it's coming from the right side, it's going through the right hepatic duct. And if it's coming from the left side, we call it left hepatic duct, right? This is all kind of coming together. It's like multiple branches of rivers starting to combine or converge. The right hepatic duct and the left hepatic duct, well, they combine together. And we kind of saw this in blood vessels, but if we take something that's big and we split it, or if we take something that's split and we combine it, but it's still a hepatic duct, well, now we're going to call it common. Great job, Ruju. Does that make sense to everybody? Does that ring a little memory bell? It's still in the liver, so it's still an hepatic duct, but we have a convergence, so it's going to be called common. Now, the common hepatic duct is going to descend. It's going to end up leaving the liver. So we're not going to call it hepatic anymore because it's not in the liver. So instead, we're just going to call it a bile duct. So that's kind of helpful, I think. Right? So down here, what you'll notice, there's the right hepatic duct, there's the left hepatic duct, where it converges together. What are we going to call this? It's still in the liver, so it's a common hepatic duct. Make sense? Now, what's going to happen is it's going to continue to descend, and it's moving down towards the duodenum. This is not in the liver anymore, so we just call it a bile duct. But it's actually going to be a bile duct that is combining two other ducts. This duct right here, which we haven't learned yet, and the common hepatic duct. So we have come together. We're going to just call this a common bile duct. That common bile duct, we'll come back to what it's going to empty into, this little outline portion in just a second. Over here on the side, we have basically a tube that leads to and from this convergence of the common hepatic duct and the common bile duct, right? Where the common hepatic duct leaves the liver and becomes the common bile duct, this is called the cystic duct. So what I'm going to do is in my flow chart, I'm going to draw an arrow that goes both ways. Go this cystic duct. This can take materials into the cystic duct or can remove material from the cystic duct. And this cystic duct is what's leading to the gallbladder. The gallbladder is a muscular sac. And I mentioned this the other day, but this is like a turkey baster. So the gallbladder is a storage container that has smooth muscles around it. And if those smooth muscles contract, volume decreases, pressure increases, then the bile is going to flow out through the cystic duct into the common bile duct. Whereas if the gallbladder is relaxing, it creates a vacuum, volume increasing, pressure decreasing, and that's going to draw bile from the common hepatic junction here into the gallbladder. Does that make sense? So now we go back to our image, starting to come together pretty well. We got our gallbladder there, got our cystic duct there. Now, the last couple of things is that we need to empty this bile down into the duodenum, but we need to make sure we're controlling it. We want it to only be released when we want it released. We don't want just a constant secretion of bile all the time that changes the composition of the fecal matter. It complicates things quite a bit. So what we have is a tiny little enlarged area. This enlarged area on this, kind of looks like a little bulb right here. These are ampulla. Ampulla might be a term you're familiar with, might not be, but an ampulla is an enlarged area that stores something. Like there are ampulla vials, ampulla of vaccines. They come in these little ampullas. I don't know if you guys have ever heard that term before. If you've ever worked in a science lab, you probably have, and if you haven't, then you probably have not heard the term ampulla before. But that's exactly what an ampulla is. Now, it's combining materials from both the liver and the pancreas. So the pancreatic juices are coming through these pancreatic ducts, descending down into this little ampulla as well. So that's why we call it the hepatopancreatic ampulla. Last but not least, we want to control what's being released from the hepatopancreatic ampulla, so we put in a small little sphincter. Smooth muscle. This is going to be controlled primarily by hormones. Common bile ducts over here. This is still in the liver. Common hepatic duct, gallbladder, cystic duct would be right there. Boom. How do you guys feel about this information? Doable? I was trying to get you guys done early-ish today, so I'm kind of finishing up here. Go ahead and you can fill in your own flow chart from the common bile duct. It's going to go into the hepatopancreatic ampulla. From there, out of the hepatopancreatic sphincter into the duodenum. At the same time, you can add in a pancreas. So add in a pancreatic flow chart, pancreas through the main pancreatic duct down into that hepatopancreatic ampulla. That part's going to be pretty easy. Pancreas is pretty darn easy. You got a main pancreatic duct and an accessory pancreatic duct. Accessory is much smaller. It's just accessory. Cool. Particularly circularis, circular folds down there. Pancreas there, spleen there, duodenum. Through this whole thing is all duodenum, and then the sphincter is that tiny little black dot that you see right there. Are there any questions about any of that? Awesome. So the last thing I'm gonna set you guys free. I'm totally setting you guys free. So I'm starting to trust that you are becoming better and better and better at studying, that you have a better foundation for the knowledge you're trying to learn. I gotta trust that at some point, right? So you guys already know you have these doc PDF files with your objectives. This is what you guys need to go through and identify on the models. This list is what you guys need to do for your extra credit. From mouth all the way down. Stomach, small intestines, large intestines, and it's going to make it to the rectum and the anal canal. So you guys can see that basically this is a flow chart from mouth to anus. So everything that we've already gone through shouldn't be too crazy, right? You'll also need to identify the different peritoneal layers. Find the pancreas, find the different ducts, the different parts of the pancreas, the liver with the different lobes, we did that, gallbladder. You need to be able to trace the pathway, hepatic ducts all the way down to the hepatopancreatic sphincter. We finished that off, and there is a flow chart here for you. Everybody feel comfortable with all of that? Yes, no? At least with the knowledge that it's there. I hope. You guys are all freaking rock stars, so I'm hoping that the answer is yes. Hard part, histology. So this is where I'm going to set you guys free. If you need me, let me know. But you guys need to be able to identify everything from the esophageal layers, the stomach layers and gastric pits and gastric glands. We have both microscopic slides, which you guys are able to go into the slide box and look up. And we have histology models. The histology models, I go and I talk to you guys about them. GI tract histology model, stomach histo model, duodenum histo model. That liver is a liver histo model. So if you guys want help with all of the models and how to identify everything on the models, I give you YouTube videos for each one of them, okay? Histo for the pancreas, histo and histo models for the liver. There's a lot of work to get done because summer. Let me know if you guys need any help. The extra credits due Friday. The extra credits due Friday at midnight. No problem. When's your guys' homework due? Saturday at 6 p.m. You guys have been doing really good at that. And then your lab practical and Unit 3 lab practical lecture exam, when's that due? I know this is dumb, but I just got to do it. Otherwise, students mess it up. Sunday at midnight. Awesome. All right, guys. Thank you. Please, please, please stay involved, study hard. Make sure you relax too. Let me know when you need help. Talk to your lab partners when you need help. Best of luck. My pleasure, guys.